Hello, hello. Hello.
Test. Okay. That seems to be working. Okay. Fine. I can't think of anything else. Good morning. Can you hear me? Okay. Hello and welcome to session 2 of Cosyne 2022. That was nice to say. I'm Cristina Savin from NYU, and I'll be chairing this second session. We have really exciting talks, one invited, two contributed, and as a reminder, we're very much welcoming questions, especially from the young part of the audience. You can either queue at the two microphones and ask your question directly, or we'll be monitoring the Q&A on the app. So if you're too shy to speak up, we'll make sure that your question gets through. Okay, so that's all with logistics for now. Since we're already a little bit late, let's get started. So the first talk of this session is an invited talk from Valerio Mante. So Valerio doesn't really need much of an introduction. There are very few people who I imagine haven't read the Mante and Susilo Nature paper on context-dependent representation in prefrontal cortex. And that work has been really influential for how the community as a whole is thinking about understanding circuits of computation through the lens of population dynamics, including the work that we do ourselves. So Valerio started his career as a physicist, followed by a PhD in neuroscience, both at ETH Zurich. He then went on to do a very successful series of postdocs at Stanford and HNI. And since 2013, he's been a group leader at the Institute of Neuroinformatics and the University of Zurich. His work combines theoretical approaches with analysis of large-scale neural and behavioral data with the goal of developing a mechanistic understanding of how cognitive processes arise through the collective interaction of large populations of neurons across brain areas. So we're very lucky to have him. Valerio, take it away.
Well, thank you, Cristina, for the introduction, and thank you, the organizers, for inviting me. So today I'll present some recent work that we've done on efforts to understand the function of prefrontal cortex in monkeys or in primates, and how these areas support cognition. And so as I think most of you know, the prefrontal cortex is thought to play a key role in executive functions and goal-directed behavior. So that's functions like behavioral flexibility, inhibitory control, decision-making, problem-solving. And we know this from a number of different lines of evidence. Also lesions in humans and monkeys, dysfunction of these areas in various psychiatric disorders, imaging studies in humans, and also neurophysiology. But generally, it's been hard to really understand the kind of computations that are happening in these areas. And one reason is that it's simply the responses in these areas are really complex, right? And it's hard to even decide what one should model if one tries to model something. And so this is, I'll show you an example of this and some findings from the areas that we mostly focus on, which are from the dorsolateral prefrontal cortex, where these findings kind of change at least my view of the kind of computations which we should include when we try to understand, when we try to build models, for example, of these areas. And so I'll start by showing you some very classic results about these areas that have really laid the foundation for everything that came after. So this is the work by Golub and Rakish and collaborators, who essentially discovered tuned persistent activity. So this is an example of a response of a single neuron in PFC. So the monkey was solving a very simple task. So monkey was fixating here at this central fixation. Then a peripheral cue was presented at one of these locations. This was followed by a delay period during which the monkey had to remember the location of that cue. And then when the fixation point turns off, the monkey makes a saccade to the remembered location. And the key discovery was that during this delay period, you find this persistent activity that bridges the time of the cue and the time of the response. And you can see this activity is nicely tuned for the direction of the saccade. And so in the coming decades, this kind of tuned persistent activity has been involved in all sorts of key functions like planning, working memory, attention, decision making, and all of these kind of support this idea that dorsolateral prefrontal cortex is involved somehow in the flexible maintenance and integration of inputs towards a choice. And this view has remained also after it became clear that really the tuning properties of these neurons are much more complex, as I mentioned. So many neurons don't have these nice tunings. So they show what is called mixed selectivity. So here's an example from our work, but there are many different examples. So what this plot is showing, each point is one neuron. Its distance from along one of these axes shows how selective it is, how tuned it is to one of these task variables. So here I'm showing 3 task variables that were important for the task that we studied, right? The choice of the monkey, the motion, and the color of the stimulus. And the bottom line of this slide is simply that for many of these units, it looks like they represent these random combinations of task variables. And again, this has made it hard to get to the bottom of the computations that this area is implementing, but insights from machine learning in particular in the last 10 years or so have really helped and shown that, in fact, we can still understand the computations. We just have to look at the population level. So these are 2 prominent examples, in particular for the areas that I'm talking about, so dorsolateral prefrontal cortex. So the work by Fuzzi et al., who showed that mixed selectivity can be exploited by these areas to flexibly categorize sensory inputs. And one can nicely understand why that is at the population level. And then our work with David, where we show that this kind of mixed selectivity emerges naturally in networks that are trained to solve these tasks. And that, again, one can then understand the computations at the population level. So all in all, looks like a success story, but it keeps this focus on decision making, right? So this role of choosing an action based on the state of the world. And the question, of course, arises, to what extent these kind of descriptions really capture the essence of, the computations happening in these areas, if not completely, but then at least, in principle. And there are actually a number of indications that there is more to the story, right? So one good example are neurons that actually respond after an action has happened. Okay, so they are not in that sense involved in the decision that led to that action. And this has been well known from long ago. So this is the same paper that I showed you before. And this is now a neuron that is tuned for the direction of the saccade, but it responds only after the monkey has chosen that option. And so this kind of post-saccadic, post-movement activity has been reported a lot in prefrontal cortex. But somehow it seems to have influenced the thinking about these areas less than this pre-saccadic activity, right, which is involved in decision making. And I think one reason that that's the case is because these studies couldn't really agree on some very important questions. First is how common really are these kind of responses? What does this post-saccadic activity actually represent, right, which is key to understand what kind of function it might play. And is it task-dependent, like many other signals in PFC? So does it emerge only during the learning of a task, or is it some stereotypical property of these neurons? And so actually, much of the talk today will be about an effort to answer these questions more precisely. And the way we'll do this is to essentially go back to the beginnings. We'll go back to a very classic oculomotor task that has a few new features, though, that will help us answer these questions. So this is essentially the same task as before, right? The monkey initiates a trial by making a saccade to a fixation point. Then a target appears at one of several locations. Monkey still has to keep on fixating. Fixation point turns off. Monkey makes a saccade to the target. Now comes the first new feature. Now there is an additional delay between this saccade and the time when the monkey actually gets the reward. And during this time, the monkey has to keep on fixating now on the target. And this small change in the task will make a big difference in the way we somehow look at the responses in these areas and will allow us to really look at this post-saccadic activity. And just as a bit of nomenclature, so I will call this saccade, right, from the fixation point to the target. The rewarded saccade is the one that is most predictive of the upcoming reward. But we'll also look at other saccades. So the start saccade is the one that the monkey makes to initiate the trial, right, to the fixation point. And then the end saccade is the first saccade that the monkey makes at the end of the trial. So this is how these saccades look, right, for one session. Again, the rewarded saccade from the fixation point to these target locations. The start saccade from somewhere on the computer monitor to the fixation point. And then the end saccade from these target locations to, again, many places on the monitor. And so we'll systematically compare the responses to these different saccades. And the third new feature is that we look at population recordings, right? So this has the advantage that we'll be less biased in choosing what neurons to record from, and maybe emphasizing some properties of the responses more than others. So these are recordings with Utah rays in the pre-arcuate cortex, right? So it's a part of dorsolateral prefrontal cortex. 96 channels. So what we get from this are about 150 to 200 units collected simultaneously, both multi-units and single units. And the way we look at these is by binning spikes and looking at firing rates, right? Time-dependent firing rates. Okay, so then here is the first result, 9 example units, right? So we're looking here at normalized firing rate as a function of time. This is aligned to the rewarded saccade, right? So the saccade from the fixation point to the target. And what we're mostly looking at here now is response during this new delay that we introduced. And so, sure enough, we can find a lot of neurons, or at least these example neurons, some of them show tuned activity for the direction before the saccade happens, right? So the colors here, the different lines are the direction of the saccade. But you can also see that there are a number of units that one can find that have this prominent tuning after the saccade has happened. And that seems to persist for quite some time. And these turn out to be representative units, okay? So we can define for each unit whether it's selective or not for direction at any given time. And so if we look before the saccade, there are maybe 40, 50% that are selective. But actually, many more units are selective after the saccade has happened. And in fact, there are really new units that come on that they become selective only after the saccade. And this is something that others had found before. There's also some interesting structure in these responses that we can see even at the single unit level. So if we focus on units that are selective both before the saccade and after the saccade, we can ask how are the preferred directions related at these two different times. So this is actually a histogram of these angular differences. And it's not flat, right? So there's not a random relationship between these times, but there's kind of an enrichment of neurons that maintain their tuning, but then also a lot of units tend to flip their tuning, which is, again, something that has been reported before. And it's an interesting feature that makes it hard, actually, to understand what these responses are, as I'll tell you in a second, and so we'll come back to this. What does this flip mean? I showed you an example before, right? So this is now an example unit that shows such a flip. It means that the responses, the directions that elicit the larger response after the saccade are the ones that elicit a smaller response before the saccade. Again, this is a very common finding. So we can look at this, of course, also the population level. So we're going to use a decoding approach. As I said, we're going to bin spikes, obtain firing rates, but now we're going to look at the entire vector, right, population vector, which has a dimensionality that matches the number of neurons that we recorded. We're going to train decoders to decode, to predict the direction of the saccade that the monkey made. Okay, so this is one example trial where this is the probability of a particular direction given the response at some time. And in this case, the decoder is correct, right? The largest probability is for the saccade direction the monkey actually performed. I'll show you results for linear discriminant decoders, but it really doesn't make a difference. You can use any other decoder. It works. The results are the same. Okay, and here is now just the same picture as before on the single units at the population level. So this is decoding performance, right? So trials correctly decoded as a function of time aligned to the rewarded saccade onset and the delivery of the reward. And again, this is long delay between them. And we can decode nicely before, but we can decode even better after the saccade. And in fact, we can decode all the way to the reward and actually even after the reward. And just as a reminder, right, so these saccades are very crisp. They're like 40, 50 milliseconds long. This is the eye velocity. There is nothing happening before and nothing happening after in terms of eye movements. So what I didn't tell you, the way we're doing this is we're actually training a decoder each time, right? So we're training a decoder and testing in a cross-validated fashion a decoder each time. So it could be that these decoders actually change, right, across time. And so we can look at this directly. So we're going to look at these very same decoders, right, which are trained at these different times. We're going to test them throughout all the times in the trial. And what we see is that there's really a lot of dynamics in this encoding. So if we take a decoder that was trained pre-saccadically and then test it post-saccadically, the performance is very small or very bad. The same is true if you train post-saccadically and test pre-saccadically. Again, the decoder is very bad. So this means the dynamics or the encoding, right, changes before and after the saccade. There are also some interesting differences in how static or dynamic the representation is before or after the saccade. This is something we've looked at in detail, and I can tell you more in the question if somebody is interested. And again, there's a lot of structure, right, in this encoding of direction. So if we, again, look at now at one of these pre-saccadic decoders, right, that was trained to predict the direction of the coming saccade. But now we tested on responses throughout the trial. That's what we're showing here again, only instead of just plotting performance, right, whether the decoder was correct or not, I'm actually showing you the actual difference, the error between the direction that was predicted or decoded by the decoder and the actual direction. Now you can see that this decoder that was trained here before the saccade does well actually at all previous times as well. But then sometime after the saccade has happened, the decoded direction flips, okay? So this is you read out the opposite direction of the one that just happened if you use this pre-saccadic decoder, and this is a manifestation of the population level of this flip at the single neuron level that I showed you. Okay, so then there is really a consistent picture, right, that emerges at the single neuron level and the population level from these kind of recordings. So a majority of neurons are selective for saccade direction before and or after the saccade, and in fact, more neurons are selective after the saccade. At the population level, this means there is this continuous encoding of saccade direction from stimulus to outcome and beyond. The selectivity of single neurons is dynamic, right? So the preferred direction can change over time. And also, the peak selectivity occurs at different times. I didn't show you this, different times in different neurons. These two effects together explain why the encoding of saccade direction is time dependent. Some neurons are selective for direction only after the saccade. So this means that at the population level, saccade direction is encoded in largely orthogonal subspaces before and after saccade. But nonetheless, the selectivity is not random, right? So many neurons have this flip in selectivity before and after saccade. And in terms of readouts, it's reflected in this flip at the level of the decoders. Bottom line, though, post-saccadic activity is really prominent feature of this area, right? And so we probably need to care about it. What all of this doesn't answer yet is what does post-saccadic activity actually represent? And so you might think that the answer is obvious, right? That it represents the direction or the metrics of the previous action, right, the previous saccade. But it turns out that there are a number of alternative explanations that would also work. It could be a visual response, right, related to the fact that the image has changed on the retina. It could reflect gaze location, so where the monkey is actually looking on the image. And it could even represent something about the upcoming saccade, a plan of the upcoming saccade. And actually, all of these possibilities have come up in the literature. And as I told you, there is not really agreement as to what it is. And so we used a number of approaches, various control experiments, but also really exploiting the simultaneity of the population recordings to exclude several of these possibilities. And I won't go into all of these analyses, but I want to show you just a few. Starting from the question whether post-saccadic activity could be the planning of an upcoming saccade. So why does this even come up as a question? And it comes up because in this task and in many similar tasks, there are these strong regularities in the behavior of the monkey. So the monkey makes a saccade to the target and then tends to come back to the fixation point because he wants to start the new trial. And so that's what is shown here, right? So this is just a histogram of directions of the end saccade for different directions of the rewarded saccade. And sure enough, you see that there's this tendency for the direction of the end saccade to be shifted by 180 degrees, right? To be opposite to the ones of the rewarded saccade. Now, if PFC is encoding the plan of the end saccade, and we know the PFC is encoding the plan of the rewarded saccade, so we can assume it also encodes the one for the end saccade. So if that's the case, then these two features together could explain many of the phenomena that I showed you, including in particular these flips that we observe in the decoders. So how do we exclude this possibility? So what we did is essentially resample our trials and try to subtract these correlations at the level of the behavior. And so the way we do this is we first pick all trials of a particular, where the rewarded saccade went into a particular direction, like these trials. And then these trials we subsample further to generate a data set where the directions of the end saccades are evenly distributed, okay? And we can do this also for other directions of the rewarded saccade. We can do it for all of them, because for some, we can't do this balancing, but we can do it for these ones. And here is just, I'm showing you first the very same analysis that we did before, right? So these are the very same decoders that we had trained on all trials. And now I'm just showing you performance on these separate data sets that we created by subsampling, and just qualitatively, the same effects are still there. We get the pre-saccadic activity, the predictive activity, and we get this post-saccadic activity. But now we're going to ask a different question. We're going to ask, can we somehow use these decoders to answer whether post-saccadic activity predicts the end saccade? So what we're going to do is we're going to pick now one of these pre-saccadic decoders, which, as you can see here, they predict the direction of the upcoming saccade. And we're going to ask, do they at some point in the trial predict the direction of the end saccade? And the answer is no, okay? So these decoders are a chance performance, and you only pick up a little bit of predictive power right before the end saccade happens. So there's really no evidence that any of this activity that we're seeing is related to the planning of the next saccade. Also, what is interesting, as you see here, there is almost no predictive activity for these end saccades, right, compared to the predictive activity for the rewarded saccade. This, again, matches actually what has been argued in the past, that you only get predictive activity, preparatory activity, for some kinds of saccades, whereas freely made saccades don't show this kind of predictive activity. But it raises a question now, what about the post-saccadic activity? Is that also gone? For these other saccades, right, so it's post-saccadic activity specific to rewarded saccades, and the answer is no. So we can, again, take our decoders that we had trained on the rewarded saccade. Now we're going to apply them around the time of the start saccade and decode the direction of the start saccade. Or we can apply them around the time of the end saccade and decode the direction of the end saccade. And you can see that at least after the saccade has happened, we can nicely decode these directions. And just to orient you here, right, so what we're looking at here, for example, this is the beginning of the trial, right? So we are like 500, 600, 700 milliseconds into the trial, and we can nicely decode the direction of the saccade that the monkey made into the fixation, to start the trial. So what this means is that really it looks like after every saccade, right, there is this stereotypical dynamics that is playing out in these areas after, again, the action has happened. And just to emphasize this again, even these flips in decoding that we observe in the rewarded saccade, they also happen for these other saccades, right? So this is shown here for the start saccade. Again, we take a pre-saccadic decoder and apply it, and you see the flip. So really, it's the same kind of dynamics is unfolding. What is different, though, as you might have noticed, is that the overall performance of these decoders has decreased. And that's mainly because the strength of this post-saccadic activity has decreased. So the its strength seems to be modulated, appears to be modulated by context, right? Where context here means the type of saccade that the monkey is making. And there are a few differences, right, between these rewarded saccades and the other saccades that we look at. So the rewarded saccade is more predictive of a reward. That's interesting. But also I showed you, the rewarded saccade is preceded by strong predictive planning activity, whereas these other saccades are not. So we can't really, at this point, say what is the key feature of the rewarded saccade that makes these signals be stronger. Okay, now, then I want to spend the last few minutes discussing, or at least speculating about what post-saccadic activity might, let me say, post-saccadic activity, what does it represent? By exclusion, it represents the direction of the previous saccade, okay? It's really about the metrics of the past action. So now, what is the function of post-saccadic activity? Again, I'm going to speculate. We don't know the answer. It could have many functions, right? I told you there are neurons that only become active post-saccadically, so those could have different functions than the one that are active pre-saccadically. And the two possibilities that I want to discuss are, again, they have already been proposed in the past. So the first one is that this has something to do with learning from past actions and outcomes. Right? So clearly, many of the actions we perform, we don't only choose them based on the inputs and the context of the current time, but it also depends on actions that we took in the past and rewards that we received. So reinforcement learning, right, deals with how to pick these actions based on these factors. But what, these frameworks don't really answer a number of questions about where these signals are coming from, for example. So for the past actions, where are they represented? How long are they represented? In what reference frame, right? Many actions, you can describe them in different reference frames, body-centered, world-centered, and at what level of abstraction, right? Is it an abstract goal or more of a movement plan? And so the results that I showed you might speak to some of these questions, right? So what I showed you is that in an area that contains plans, in terms of saccadic plans for visual goals, in a specific reference frame, right, retinotopic coordinates relative to the fixation point, we also find what look like memories, right, of the past action in the very same reference frame. And we can, this is a conjecture at this point, maybe this is something that one finds in other areas as well, right? So we know that there are other parts of prefrontal cortex that represent plans to much more abstract goals in more cognitive spaces, and the question is whether maybe there one could find memories now for past actions that were based on these plans in these very, very same cognitive spaces. So that's something to be tested in future experiments. And now the second possible function, which again has been discussed a lot in the past, is related to updating existing plans based on past actions. And so this is actually a figure from a very early paper, Goldberg and Bruce, 1991. You probably can't read most of the things on this slide. So essentially it shows how the frontal eye fields contribute, right, so to oculomotor behaviors, right? The frontal eye fields, which is a part of the brain very, very closely related functionally, anatomically to the areas that I described. So it receives inputs, right, from visual pathways. It transforms these inputs somehow into plans for saccadic movements, which are sent to a brainstem saccade generator, which then moves the eyes. And post-saccadic activity actually comes up in this model. So I'm going to zoom in here. So here's post-saccadic activity. It's driven by an efference copy from this saccade generator. And it interacts with this transformation of inputs into plans with this minus sign, right, this subtraction. So what is being subtracted here? Again, this is based on a suggestion by Bruce and Goldberg and others. So imagine that you're fixating here, right, at this particular location in the image, and now you become aware of two other salient regions in this image, and so you want to plan a saccade to these regions. So those are represented, these plans, as these vectors, right, relative to the gaze location in these areas, like the ones we record from, which employ retinotopic coordinates. Now, after you have made the first saccade to this first goal, you have to modify the plan that you had originally, right, to make a saccade to reach this second goal, right? So the new plan that now applies from this case location is this one, this plan B star. And so what is plan B star? Well, it's just, vector addition. It's simply plan B, the original plan that you had, minus the action that you just performed, right, minus action A. So this is, there have been many suggestions that the brain is doing something like this, right, between saccades. And now there is this intriguing observation that one could argue that we actually observe in these areas a representation of this minus action A, right, the opposite of the action that the monkey just took in terms of these flips, right, at the level of the single neurons and at the level of this pre-saccadic activity, right, the change of the readout that we observe after a saccade. Now, I'm not saying that this finding now means that this kind of computation is happening in these brain areas, but it's at least an intriguing possibility. And it certainly, though, led us to think that we need to include some of these computations in the kind of models that we are generating, right? So the idea is not to give up on the idea that these areas are involved in decision-making and the work, that we've done in the past, but to implement these decision-making processes in a setting that somehow reflects the constraints that the visual system is actually operating in particular the fact that the visual system is actively sampling information, right? So here is a version of such a model, right, which has to classify MNIST digits, right? There's a part of the model that does this classification, but now the key feature is that the model only sees a very small portion of this image at any time, so this would be the fovea, and has to sample this image by actively generating saccades, right, and moving this fovea around. And then has to accumulate information from these different saccades to achieve a good classification. And again, this is how the visual system usually operates, right? And we can certainly train such networks, right? They generate policies that then sample these images in a meaningful way, and as these models sample, they can integrate evidence and improve classification performance. And so, our hope is that, again, by combining these two features, right, the decision-making and active sampling, we might explain some of these really prominent features of the responses that are outside of the scope of the models that many of us have developed in the past years. Right, so to summarize then, prefrontal cortex maintains a continuous representation of saccadic actions, right, from the plan to the outcome of these actions and beyond. In this area, this happens in a fixed coordinate system, right, the retinotopic coordinates, with dynamics that are independent of saccade type, but with a different strength for different saccades. And we don't know, again, what is the critical feature that influences the strength of these signals. So, and I think this is some of the best evidence so far that maintaining representations of past actions may be a key function of dorsolateral prefrontal cortex. And I speculated about the functions of post-saccadic representations, right? I discussed these two possibilities. Really, to distinguish these, we need better experiments, so where we can really probe on a trial-by-trial basis the correlations between these signals and behavior. And as I showed you, I think we also need better models in the sense that we need models that include more natural tasks and more natural constraints of the kind that, right, the visual system deals with. These are the people that did the work, and the analysis and the experiments and the funding sources. Thank you.
That was great. Remember that you can queue. All right, somebody's doing it already. Go ahead.
Hi, Valerio. I'm Megan Carey from the
Hi.
Champalamo right down the road. That was a really nice talk. And I'm wondering what's known about this kind of post-saccadic activity in other saccade-related areas, like either other areas in the cerebral cortex or subcortically, like the superior colliculus or the cerebellum, and whether there are differences that might give us hints as to the computations in these different areas.
Right. Yeah, so I can mostly say something about, other prefrontal areas, right? So so if you go back to the literature, it turns out that even in the frontal eye fields, right, which is really the output area, right, that directly projects to the superior colliculus, even there you have post-saccadic activity, right, that has many of the same properties that I described. It's there, you know, as you go more anteriorly, and interesting, at some point, there is an indication of this even in our recording, that in arrays that are more anterior, you start losing the planning activity, but you still keep the post-saccadic activity. And in fact, when you go to frontopolar cortex or orbitofrontal cortex, similar experiments have suggested that the only thing that is left for this kind of saccadic tasks is post-saccadic activity. So those experiments, though, tend to be more impoverished, right? So it's this decision-making task where you only have two options, and it becomes very hard to understand what is the thing that is being represented. And I think there, it's easier by having this higher dimensional motor output. To what extent they exist in other areas, I think it's hard to say at this point. People report responses after an action, of course, but interpreting it and what exactly it represents is really hard in many experimental settings.
Well, I'd love to know what you would expect to see, but we can maybe chat offline.
Cool, great.
Okay. We're going to take a second question from the online crowd, from Chris Curran. I'm quoting here, great talk. This is probably a very naive question, but would a double saccade task, one after the other, so where I think there's a sequence of saccades that you need to do in order to achieve the goal, give us potentially more information about the role of post-saccadic activity? And do you think your results would extend to this?
Yes, so the double saccade task is one that, you know, where a monkey is presented two targets in a sequence and then has to remember, oh, I need to make a saccade to the first target, and then I need to make a saccade to the remembered location of the second target. And there is a lot of interesting literature on how, and this task has been used as evidence that monkeys use a corollary discharge, right, from the saccade generation system to up their plan of the second saccade. So yes, I think similar kinds of experiments could be useful also in looking for these trial-by-trial correlations that I've been referring to. Yeah.
Great. Go ahead.
Hi. Have you considered physiological versus functional explanations for the data, like maybe the synapses involved or the neurons or synapses involved in the pre-saccadic activity just become depressed during the post-saccadic activity, and even if it's not playing a functional role?
I don't know. I mean, what is striking is this activity really remains for a very long time, right? So these delays, they didn't explore, you know, with more than maybe 1.5 or 2 second delays, right, between the action and the reward, and this activity is still there. I showed you, you know, it extends even further, right, into the beyond the reward. So I don't know. Certainly I showed you that there are new neurons, right, that become selective only after the saccade has occurred, right, that show no pre-saccadic selectivity. So I'm not sure how one would explain that with a mechanism like that.
Go ahead.
Thanks. Hi, Valerio. That was a great talk. I had a question about the timing of the responses pre versus post. So if I got it right in your decoding, with your decoder, it looked like there was more homogeneous sort of decoding across time pre-saccade, but afterwards.
Yeah, exactly. Yeah. So this is something I didn't show. These are kind of small cutouts of these matrices, right? So this is before the onset of the rewarded saccade, and everything looks nice and homogeneous. And this is early after the rewarded saccade here. And this is aligned to the reward delivery. And both of these look less homogeneous, right? More dynamic. And we actually looked at this, you know, very carefully, right? So we can really get a number on how dynamic a particular single trial is, right, in terms of encoding. And there's this really big difference, right, between how direction is encoded before a saccade, where you really have this very stable encoding, and the way that direction is encoded after the saccade, where you have some trials that are look like static and some trials that are very, very dynamic. But overall, you have a much more dynamic encoding. So yeah, this is another very interesting difference, right, between these two, between the pre-saccadic encoding and post-saccadic encoding, really suggesting these are different signals, right?
Yeah, very, very exciting. So for the post-saccade, is it the case that individual neurons have peak latencies at different times after the saccade?
Yeah, so that's one effect that contributes to these dynamics. The other effect, as I mentioned, is that the preferred direction can change over time, even within single units. And you kind of need both effects, the different latencies, right, of the peak selectivity and the change in dynamics within neurons to explain these kind of effects.
Thank you.
Go ahead.
Hi there, nice talk. My question is about the speculation part of your talk.
Mm-hmm.
So I think I was able to follow and found it really interesting, your discussion of credit assignment, this idea that post-saccadic activity would sort of be online so that credit assignment could then take place, maybe through some kind of a prediction error computation. I followed that. Then you kind of went on to discuss the info sampling aspect, and I was curious how you're sort of thinking about that, if you could sort of expand the link of post-saccadic activity and DLPFC activity and info sampling. I wasn't...
In what? Sorry, what sampling?
You were talking about information sampling,
Active
right?
sampling, yeah, exactly. No, so the active sampling, so the idea is that you have a fovea that you move around, and what you really care about is gathering information about the outside world, right?
Got that.
And it's unclear whether, for example, whether the saccade system is employing information about where the eye is relative to the head. And there's actually good evidence that it's not using that on short time scales, that the only information it has to keep track of world reference frame is the information about the saccades that it just generated, right? The action that it produced in these relative coordinates, right? In retinotopic coordinates. So you need to do something like, you need to integrate these deltas, right? These actions into a global picture of an image in a global reference frame. And so the suggestion, right, was that post-saccadic activity is used for that process, right?
Thanks.
We are running out of time, so let's thank again our speaker for a great talk.
Thank you.
And we'll switch to the first contributed talk of the session from Jordan Elum.
Okay, I'm Jordan Elum, and thanks for the opportunity to speak. So today I'll be talking about learning and motivation-related signals in dopamine neurons, and so I first want to start by highlighting one of the foundational studies that implicated dopamine neurons in reinforcement learning. So we know how dopamine neurons respond to rewards and cues thanks to these single unit recordings from the primate midbrain. So this dopamine neuron is activated by an unpredicted fruit juice reward. It fires more action potentials right after the reward is delivered. But after learning that a predictive stimulus precedes the reward, the dopamine neuron is activated by the cue and not the reward itself. And then if the reward fails to occur after the predictive stimulus, the dopamine neuron activity is depressed at the time that the reward was predicted. So this led to this model that says dopamine neurons compute a reward prediction error signal, which is the difference between expected and received rewards. So around the same time, another set of foundational studies implicated the dopamine system in motivational processes. So in these studies, the experimenters predicted that neurochemical lesions to the dopamine system would negatively impact positive affective reactions for food reward. But instead, what they found is that disrupting the dopamine system in rats actually abolished all motivation. So the rats were no longer interested in seeking out or consuming food rewards. So this in part led to this incentive salience model of incentive motivation, which states that dopamine neurons are needed to generate wanting or to attribute incentive salience for cues and actions related to rewards. So one of the major questions that I'm interested in is, do all dopamine neurons perform these functions uniformly, or do distinct populations of dopamine cells contribute to the two processes? So technological advances in the past 2 decades have led to a much greater understanding of heterogeneity in the dopamine system. So we know that dopamine neurons reside in several groups in the midbrain and then project broadly throughout the brain. And techniques that allow for defining dopamine neurons based on their downstream target can really greatly enhance our understanding of their role in behavior. In addition to that, techniques that allow us to record from anatomically and molecularly defined dopamine neurons have already begun to reveal many unexpected response profiles. So for example, in the VTA of mice navigating a virtual reality maze while performing a complex decision-making task, we see that dopamine neurons are actually representing 1 or 2 behavioral variables, such as the trial accuracy, reward history, spatial location in the maze, all in addition to reward outcome encoding. So these findings have already begun to challenge this classic view that dopamine neurons are uniformly broadcasting learning or motivation-related signals. So one of the most striking examples of heterogeneity in the dopamine system is the variability of projections to distinct regions of the striatum among subpopulations of dopamine neurons. So the striatum integrates these dopaminergic signals with cognitive and sensory glutamatergic inputs. And unsurprisingly, striatum is essential for reward association learning and also for generating motivated behaviors. But one subject of debate in the field is, what is the role of VTA dopamine release and projections to specific subregions of the ventral striatum or the nucleus accumbens? And so one hypothesis that was originally proposed by Ann Kelly states that dopamine release in the core facilitates the learning about motor actions that led to a reward, while dopamine release in the shell of the nucleus accumbens provides an incentive salient signal and promotes behaviors that will help bring an animal in contact with a rewarding stimulus. So to test this 2-component neural circuit model, a former graduate student in the lab genetically isolated core and shell projecting dopamine neurons. So core projecting dopamine neurons express CRHR1, and shell projecting dopamine neurons express CCK. And then he wanted to model reward association learning in mice, so he used an optogenetic intracranial self-stimulation task with the excitatory opsin channel rhodopsin expressed in either core or shell or all VTA dopamine neurons. And unsurprisingly, DAT-pre mice self-stimulated for activation of all VTA dopamine neurons, and also phasic activation of the core projecting dopamine population was sufficient to promote learning, although not in the shell projecting population. But interestingly, after training mice on lever press conditioning for food reward, the stimulation of the shell projecting population was sufficient to enhance the effort or motivation level of the mice for stimulation, even though the stimulation was not reinforcing initially. So these results are consistent with a model in which dopamine projections to the core regulate reward association, where projections to the shell regulate motivation. So these results kind of led us to ask whether we could detect distinct learning and motivation signals in the endogenous activity dynamics of these neurons. And more specifically, does activity in the populations diverge at action, cue, and outcome? And can we detect evidence of prediction error or incentive salient signaling in either or both populations? So I used a similar genetic strategy to isolate core and shell projecting dopamine neurons, and I selectively expressed GCamp in the VTA of these Cree mice, and then I used fiber photometry to record the intracellular calcium dynamics as a readout of neural activity during different phases of instrumental behavior. So I first used a cued reinstatement task with acquisition, extinction, and reinstatement phase to assess activity dynamics during this task. So during the acquisition phase, an active lever press starts a 3-second delay period, followed by a 3-second audiovisual cue, which is immediately followed by delivery of a sucrose pellet. And mice acquire the lever press response over 5 days and strongly prefer the active lever over the inactive lever. And so, unsurprisingly, all VTA dopamine neurons in these DAT pre-mice expressing GCamp in the VTA, the neurons are activated by the lever press and the cue and the reward. Although when we align the activity in the core and shell projecting populations, we see divergent activity dynamics during the same behavioral epochs. So in the core projecting cells, we see the same phasic activation pattern, although the activity in the cells returns to baseline between the discrete events. And that's in contrast to the shell projecting dopamine cells, which maintain the increase in activity throughout the action, cue, and outcome periods. And so that's all on the first day of acquisition. And then on the last day, we see a similar pattern of activity, although the difference in activity between the populations following the lever press is greater. So to see what the behavior looks like, aligned to the photometry signal in one trial. So first, we're looking at a CRHR1 Cree mouse to isolate the core projecting cells. The mouse does a lever press. Cue turns on. The reward is delivered, and we get increase and decrease to each of those events discretely. It does 2 more presses. We get 2 more peaks for that. So if we look in the shell projecting cells, mouse did a lever press, cue turns on, reward is delivered, and we get increases in activity, but the activity has not come back down to baseline for a full 10 seconds after the reward was delivered. So following acquisition, we train the mice on the extinction version, and so that effectively both levers are inactive, and there's a cue and reward emission. The mice extinguish a lever press response, and we see a small increase following the lever press, but no difference between the populations for cue and omission encoding. So following extinction, we do reinstatement in a single day, where the original reward predictive cue is presented again, but that's followed by reward omission, and the mice reinstate their lever pressing for the presentation of that cue. And we see no difference following lever press between the groups in activity, but as we saw during acquisition, the shell projecting cells maintain their activity during the cue and have greater activity during the reward omission period. So next we asked if the activity in these populations is sensitive to reward probability or expectation, and also whether we could find evidence of negative prediction error encoding during unexpected reward omission. So to test this, I modified the original acquisition tasks to omit the reward on 25 or 50% of trials on consecutive days. And so what we found is that both populations showed decrease in activity following the reward omission, but only the core projecting population dips below baseline. And then during unexpected reward delivery, we see that both populations increase their activity with increasing uncertainty of reward delivery, although this effect is stronger in the shell projecting population. So finally, we then asked if either projection population is sensitive to the level of effort or work required to obtain the reward. So I used the progressive ratio task in which the lever press response requirement increases throughout the session to determine the amount of effort an animal is willing to work for a reward. And so you can see in this lever press ethogram that this generates a high rate of lever pressing up until breakpoint. And so what we found is that following the lever press and also during the reward retrieval period, the shell projecting population had increased activity compared to the core projecting population. So this was kind of a similar effect to the changing reward expectancy we saw with random reward omission between the two populations. So to summarize the main finding, we found distinct activity dynamics in core and projecting VTA dopamine populations during instrumental behavior. So the core projecting neurons provided temporally discrete information about actions, cues, and outcomes, possibly to provide predictive signals and facilitate value-based learning, whereas the shell projecting dopamine cells provided sustained signals throughout all these events, possibly to facilitate the attribution of incentive salience to specific actions and cues. And so these results provide further evidence that the midbrain dopamine system contains functionally distinct subpopulations for the regulation of learning and motivation. And in the future, we're interested in looking into mechanisms that might underlie the different dynamics in vivo, but most importantly, in integrating our findings of these associable learning and motivation signals into a more cohesive framework of dopamine function and behavior. With that, I want to thank all the members of the Zweifel lab at UW, my collaborators, the UW Graduate Program in Neuroscience, my thesis committee, and funding sources.
Thank you, Jordan, for a great talk. We have time for maybe a couple of quick questions.
Hi,
Go ahead.
thanks. I'm Ethan Bromberg-Martin from WashU. Very nice talk. I was wondering, since you said that the dopamine in the shell could influence performance on the progressive ratio task, and you're highlighting sustained activity, I'm wondering, could you use activity during leading up to or during lever presses to predict something about their behavior? Like maybe there's more dopamine in the shell when they press more avidly?
Yeah, that's actually something we're interested in. I didn't quantify it or show it in this talk, but we see kind of evidence of ramping in the shell projecting dopamine population leading up to the action, but we don't see that at all in the core projecting population.
Okay, very cool. Thank you.
If there aren't any urgent questions, then in the interest of time, let's thank Jordan again and move to the last part of the So our last contributed talk of this session comes from Karyna Mishchanchuk. I butchered that one. Karyna, take it away.
Hello, everyone. I'm really excited to give my talk here. And as part of my PhD at UCL, I'm really interested in how mice behave in a constantly changing, uncertain environment and what strategies they use to guide their behavior and to maximize their reward. And I'm excited to give a brief introduction to what I've been working on so far. So for this, to study this problem, we use a two-arm bandit task, which is a probabilistic reversal learning paradigm that's used across different labs to study this kind of problems across many different species. And in this task, the animal, the mouse, has to nose poke to initiate the trial. Then it's presented with two levers, and it has to make a choice to press one of these levers to get Sweetwater reward. And in a given block of trials, one of these levers would be the high probability choice, so it will give reward 70% of the times the mouse makes that choice. while the other lever would be a low probability choice, and it will be yielding reward 10% of the trials. So then the mice would repeat this for a few trials until the reward contingency is reversed, and the identity of the high probability choice will change to another lever. And because of the probabilistic nature of the task and the continuously changing blocks of the reward contingencies, for optimal performance, the mouse has to continuously integrate its past trial outcomes to infer the identity of the high probability choice. And what I think is very interesting in this task is that this can be achieved with many different behavioral strategies, and potentially the choice of the strategy may mean the different neural bases that would underlie the behavior. And that's why I think it's very interesting to study this problem. So first, I'll show you the example session. I don't know if you can see there are gray boxes that identify the blocks where the right lever is the high probability choice. They're like around there, but I don't know if you can see it, sorry. So the blue dots indicate when the mouse receives reward from right or left lever press, and the trace is the running average of mouse proportion of the right lever presses. So I don't know if you can see the blocks, but this basically shows that the mouse choices actually follow the reward blocks very accurately. And this is also illustrated here, where I've plotted the proportion of the high probability choices around the trial where the reward contingency is reversed. Again, this shows that the mouse is very quick to pick up on the reversal and start choosing the new high probability lever more often than the previously high probability lever. And all this means that mice actually get really good in solving this task, and I think it's really impressive for a mouse to become so good in this task. And the main question we had was what strategies they use that allow them to perform so well. For this, we turned to the behavioral modeling, and we looked at the two families of models that are most commonly used to model this simple mouse behavior. First, this is a value updating strategy. That's just the basic Riskola-Wagner rule, where the expectation of the choice, expected value for the choice, is updated with the reward prediction error scaled with the learning rate alpha. And this is most commonly used to model rodent behavior. While in human and primate studies, the models that are most commonly used are state inference models, where the subjects are expected to use the observations, so choices and their outcomes, to infer the probability of current state of the task. And in this task, the states are it's either the right lever is the high probability lever or the left lever is the high probability choice. And then we were interested to see how these models describe animals' behavior and what model fits better. So we looked at the different number of these models with loads of different combinations of different parameters. And for each mouse, we picked the best fitting model from each of these families, so then we could compare just two families. But for all of the mice in both of these model families, the winning models always had the reward and punishment sensitivity factors, and also bias and perseverance factors sometimes. So we've compared these models, and here you see the plot of the delta AAC, which is a difference in the goodness of the fit from the best fitting model. And overall, the state inference models provided best fit for all of the mice for this behavior. And then also in the simulation from these models, overall, the simulation from the state inference data model provides better fit to animals' behavior around the reversal overall. Then we were interested in what makes the state inference model better than the value updating model. And our suggestion was that because of the symmetric nature of this behavior, the main advantage of the model, of the state inference model, is that it implies the interdependence of two choices. So when the probability of right lever being the high probability lever goes up, the probability that you're in the left lever block would go down. And to test this, we've modified the value updating model to utilize this property and to perform the double updating of both choices on every trial in opposite directions. And indeed, the double updating strategy gave better fit to animals' behavior, and also the simulation from this model recapitulated behavior even better. So what I've shown so far, that during the probabilistic reversal learning, mouse behavior is best fit by models that incorporate state inference or double value updating, and this is probably due to relational lever representation in these models. Next, we wanted to see whether the neural representation of behavior is supporting our hypothesis that mice perform relational updating. And for this, we've generated predictions based on the trials where mice change their choice from A to B. And they're very simple predictions. I'll walk you through those. So if the mouse makes choice A and gets the reward, the value would go up. And if it doesn't, the value goes down. And for the choice B, if there is no double updating, the value stays the same regardless of what happened before. And then it makes the choice B and gets the reward. And the reward prediction error is the same regardless of what happened on the previous trial. However, if there is double updating, the outcome of choice A will also affect the expected value for choice B. Therefore, by the time the mouse makes choice B, there will be different expected value for that choice. And therefore, when the mouse makes choice B and gets the reward prediction error will be different. And it will be higher following A plus B plus trials compared to the A minus B plus trials. So overall, this suggests that depending on the behavioral strategy the animal uses, there will be different reward prediction error based on what happened on the previous trial. And to test whether this is supported with the neural representation of the behavior, we turn to recording midbrain dopamine, because it's often used as a readout of the reward prediction error. So for this, we used the dopamine sensor D-Lite and recorded dopamine release into the nucleus accumbens. This is example trace, and we could see the dopamine response at the time of the reward. And we could also split these traces in the same way I talked in the previous slide. So as you see, the dopamine response following A plus B plus trials is much higher compared to the A minus B plus trials. And this was also consistent across mice and in agreement with our prediction from the double updating or state inference strategy. So what I've shown so far is that during the probabilistic reversal learning, the mouse behavior is best fit by the models that utilize relational updating. And the dopamine release in the nucleus accumbens during this task is consistent with relational lever updating. This was a very specific small example. I've also done more general regression analysis where I've shown that with that error from the state inference model provides much better prediction of the dopamine response compared to the reward prediction error. Just don't have time to show this, but happy to talk about this later. Next, we were wondering what's the source of the relational representation, and in literature, there is a number of regions that are highlighted as the source of such computation, and including frontal cortices and hippocampus. And I'd like to spend the last minute of the talk to tell you why we think that hippocampus might be important for this behavior. So we've repeated the dopamine recording experiments, and also at the same time, we removed the pyramidal neurons in the ventral hippocampus unilaterally with the caspase construct expression, and in the control group, I've injected a fluorophore in the same region. And as you've seen previously, depending on the past trial outcome, dopamine response is different in the control group. However, in the caspase group, there is no such effect. And again, this was consistent across mice, and the data from the unilateral caspase group was more consistent with our prediction from the regular simple value updating model. So overall, this suggests that consistent with the role of hippocampus in relational learning and updating, mice that had their pyramidal neurons in ventral hippocampus removed with the caspase construct unilaterally didn't show evidence of relational updating in their dopamine traces. Okay. And the main summary for the talk is that during the probabilistic reversal learning, mouse behavior is best fit by the models that utilize relational representation, and dopamine release in nucleus accumbens during this task is consistent with relational lever updating, and neural representation of this behavior depends on activity in ventral hippocampus. Again, I'm short on time, so I can't talk about other experiments I've done to show the role of hippocampus in this behavior, but happy to talk about that later. And the last slide, I'd like to thank the Wellcome Trust and the Vickers Family Trust for funding my PhD, the MacAskill lab members, and huge thanks to Andrew for his support and supervision. Thank you.
So we have time for a few questions. While we're getting started, I'll read one online. Thanks for the great talk. In the first section, it seemed like the double value updating was better, but still not as good as sort of doing proper model-based reinforcement learning and state inference. What other properties of state inference might mean that it performs the best?
This could
what's
also
missing?
Okay. Yeah, this could also be due to the fact that the double updating model just has more parameters. It has 2 more parameters than the state inference model, and it's been punished by the model comparison. Yeah, I'll try other. We have an idea of how to do it with the fewer parameters, so it might even improve it more. But yeah, the main idea is that it's better than the regular value updating, even having more parameters than that one as well. Thank you.
Nobody in the audience? I'll ask a question then. So can you say a little bit more about how you were thinking about the hippocampus's role in the inference process, at the computational level, perhaps?
Sorry, your question is why we think hippocampus is important?
Yeah.
Yeah, I guess it's in agreement with a lot of Eichenbaum lab studies with the relational updating and learning, but also you can think about the 2 blocks of the task, so right or left lever being the best choice, being 2 separate states, and there is a lot of theory around that hippocampus is representing the state map of the world of the tasks, even if it is not a physical map. So it's possible that in this task, that's the role it's performing.
Mm-hmm. But I thought that it's primarily for model-free reinforcement learning, not so much for model-based. That's what I was curious about. Also, is there a particular subregion of the hippocampus, or this was pretty broad?
This is all done in the ventral hippocampus because that's the region that directly projects to the frontal cortices and the nucleus accumbens, and that way it might potentially affect the dopamine response because those regions project to VTA.
Great. Looks like we have another question.
Hi, really nice talk and data. So Jane Wong's meta-learning model can reproduce this kind of relational learning by basically encoding the state of the task through a recurrent network of LSTM units. And in that paper, they sort of proposed that recurrent network is sort of embedded in prefrontal circuits. Can we think of your data as supporting the idea that something like that kind of recurrent processing to encode the state of the task might actually be embedded in hippocampal circuits and thus kind of approximate the kind of state inference in your more statistical model?
So what was the question?
Can we think of this state inference as being basically accomplished at an algorithmic level through kind of recurrent processing in circuits that aren't in prefrontal cortex but actually are embedded in hippocampal circuits? So how do you think about mechanistically? Do you think that model might be a good model for what these ventral hippocampal circuits are doing in the context of this task?
I think so. So because there are theories about the role of hippocampus in the you said recurrent circuits, right? So it's possible. I don't know about the mechanistic theory of the circuits of the hippocampus, but I think it's plausible that it's happening there. But it's clearly whatever the hippocampus is sending off forward to PFC is useful for that computation, even if that's happening in PFC or hippocampus.
I just think it's potentially cool that you have a mechanistic model out there that could underlie the kind of statistical model that best fits your behavior. It would be interesting to talk about it offline.
Yeah, thank you.
One last question.
Yeah, those are somehow two, and I'm not sure which one is stupid and which one is okay. So what I'm thinking is that if you have many choices, then the coupling between lowering the reward of one and updating the reward of the others would be minimal, right? So when I think of like mice in the wild, then I would assume that they have more than 2 choices. So the question comes somehow to ask, the question I want to ask is whether you think that this somehow relationship between the two sides and 2 rewards is something that is intrinsic to how the mice think, or if it's something that is taught to them by the task, because they somehow have a hard time imagining that this is how the wild behaves.
Yeah, this is very specific to the behavioral task, of course, because they're taught that there are two options and they're interdependent, and when one is better, the other is not as good. But I think overall it's also important to show that with the task they do and with the way they solve it, they are able to have the representation of multiple different states and have the way of connecting them to make the best choices. And even if there are more than two states in the world, obviously they are able to weigh them relative to each other and understand the outcomes that lead from them.
Okay, I mean, okay, yeah, sure, yeah, well, yeah.
Yeah.
It's just that multiple choices would decrease the rewards that you would expect from any one with respect to one very fast. So it's not so much that you can decrease one by reading another. I mean, when you have many choices, that would disappear very fast. So
maybe it's not as drastic in the real world, because this is the state. This is the task with just two states, and that was the most useful strategy for them to solve it.
Okay, yeah, fair enough.
Yeah. Thanks.
So that's it with the questions. Karyna has a couple of words that she wants to say before we wrap up this session.
Yeah, I just wanted to say something that's personally very important to me, because I am from Ukraine and I have loads of friends and family who are still there. And I just wanted to use an opportunity talking to such large audience to say how it's still super important to keep talking to everyone around you about this, because I know it's been 3 weeks since everything started in Ukraine, and it's, I think it's just very important to keep talking to your governments, to your employers, to ask them to do as much as possible, and also to, personally, I'm sure most of you have already done so, but to donate to many of the charities that are actually doing a lot to save lives of people in the country, and over 3 million of people who've already left. But if anyone is still uncertain and wants to talk and ask me what the charities I think are great, I'm also happy to answer that question. Thank you.
Okay, so that ends session 2. We'll be back at 10:45.
And what happened? You're not sure?
I think I can just look at this time here. Thank you. Okay. How do I turn this on? Okay. Hello, testing, testing. Hello, everyone. Welcome to session 3. Let's get settled in. Just one announcement before we start the session. Due to COVID, of course, we do not want anybody to take off your mask in this auditorium, so please refrain from bringing any food or drinks in here and eating them inside this auditorium. Okay, so with that, I'm very excited to start this session. The session is on communication within and between brains. I'm Memming Park. I'm going to be chairing the session. And we have very exciting talks. We have 5 talks total, and then we'll have a lunch break. All right, so our first speaker is Michael Long. He did his PhD in Brown and then postdoc at MIT. He's currently a full professor at NYU, where he's been a faculty since 2010. And his CV is too long for me to read, and I lost my phone, so I can't really give you a long introduction. But here's the great Michael Long.
It's amazing to be here in person, and I think many of us have had the natural experiment over the last 2 years, really almost to the day, of being really hampered in the way that we can communicate with others. And I think everyone in this room has the same kind of Zoom fatigue that I do. And I think it really brings home how important it is to interact with other people. And this, of course, can be really devastating in cases of communication disorders. So this is a person that has aphasia. I should tell you, communication disorders affect about 15% of the population. And this is a person who is a computer technician. And then after having a stroke, he really, his ability to communicate has been reduced to just a few very simple words.
And what happened? You're not sure? Did you have a stroke?
Yeah.
Okay. And what symptoms did you have? What's wrong with you now? Okay, so you can't move the right side of your body. Are you able to speak okay?
Well, yeah.
Okay. Do you feel that your speaking is normal?
No.
What did you used to do for a living?
Yeah.
Can you talk about it? You're pointing to the computer. I guess you work with computers, is that right?
Yeah.
Just talk about it a little bit if you can.
No.
You can't?
Yeah.
Does it bother you having trouble speaking?
Yeah.
So it really makes you think about the act of speech. And talking is something that's actually remarkably complex when you think about it. So words have a rapidly changing acoustic structure on about a 20 millisecond time scale. Each phoneme requires that you move articulators in a very precise way in order to produce intelligible speech, and this involves about 100 different muscles. And of course, we're not all speaking into the void. We typically use this speech to communicate with other people. And so this is an annotated portion of a podcast where this person in white is talking. You can see the segments of his speech, and her speech is shown here in gray. And you can blow this up and see one of the hallmarks of interactive speech is this kind of turn-taking back and forth. People have looked at this, quantified this, including Levinson, who's at Max Planck in the Netherlands. And the time it takes for one person to respond after the other person talks is typically extremely short. So if we think we blink our eyes, and this is about 200 milliseconds, that's the same amount of time on average it takes for us to respond to our partner in interactive speech. So how does this work? Right, what are the processes going on in her head during this kind of spoken interaction? Well, people haven't really formalized any models about interactive speech, and so we decided to give it a try in a review that's coming out in a few months. And the idea is this. Of course, in her mind, she's going to have to parse this sensory stimulus, which is the other speaker. And then she's going to have to make her own speech, right? This kind of motor processes. And these kinds of things have been looked at by a lot of really incredible work from Eddie Chang and a number of other people. And I think this has been, and Krishna Shenoy, and this is something that is really knocking on the door of things like speech prostheses, which is, I think, going to be an incredible advance for society. But beyond the sensory and motor side, we really don't know much about the cognitive processes that are also really critical for this. So I sat down with Greg Castellucci, who's a postdoc in the lab, and Frank Gunther, who's a speech modeler who works at Boston University. And we tried to put together a series of different modules, processes that have to be to happen in order for interactive speech to occur. And this is based on two things. It's based on decades of linguistic research, and it's also based on what happens in the clinic. So specific aphasias that can affect specific processes in this interactive speech context. So there's content, right? So if I hear what you're saying, I have to formulate a response, and that involves several steps of getting together my answer. There's timing, right? So this has to be deployed at a very specific time in order, for example, that kind of turn-taking to occur. And then there's more of an affect side. So this is going to be colored, of course, by our emotional state. And in some cases, affect can directly drive movement. So if you think of crying or screaming, these more emotional vocalizations can be directly driven by this affect pathway. So as I mentioned, we know very little about how the brain does this using the kind of more precise tools that have emerged. Things like Eddie Chang has done using electrocorticography to try and see how the brain is doing this with high temporal and spatial precision. So we decided to take a whack at this module, which is response planning. Specifically, we're interested in the planning processes required for interactive vocal communication. And for that, the person who's really led this effort is a very talented postdoc in the lab, Greg Castellucci, and a team. We've been collaborating with these guys in Iowa for about a decade now. And Jeremy Greenlee is the one who's the principal collaborator, as well as his larger team of different neurosurgeons and support staff. So the general idea is this. There's electrocorticography, so these are sensors placed directly on the brain of patient volunteers that are undergoing neurosurgical treatments for medically intractable epilepsy or brain tumors. And I should tell you that everything I'm going to show you today we see in both of these patient populations. So it's not something that's selective, for example, for one underlying problem or the other. There's high spatial resolution and temporal resolution on a 10 millimeter and 10 millisecond time scale. And what's amazing is that even in this very challenging context, interactive speech looks pretty normal. And so this is just a conversation that we've annotated between an experimenter and a patient. So we've taken all of the things that the experimenter says and just aligned to the offset of that speech and looked at the patient picking up and answering in conversation, and you can see that this is happening with that same sub-second latency. Okay, so we wanted to isolate the planning processes to get at this kind of interactive speech planning. And to do so, we used an extremely clever task that was developed by Sarah Bogles in Stephen Levinson's laboratory. And it's called a critical word task. And let me very quickly walk you through what that is. So the idea is there's a series of questions, and there are about 60 questions that we administer as part of this task. And there's a critical word in that question that can lead to the answer. The rest of the question is really kind of filler, context. And that can then initiate the response planning. I'll give you an example. So body parts, how many fingers does a healthy person have? Toes, arms, legs, et cetera. We had to translate this from the original Dutch, which was really our only contribution. And antonyms, and then just to keep things kind of fun, we have animal sounds as well. Moo, oink, what does a fox say, I guess, you know? And then we see responses that look like this. So just to orient you, there's an array that sits here in the brain. There's a strip of electrodes that's down here in an auditory region, actually from a different patient. But we can align this thing to the structure of this task. and I'm going to show you the average responses in a 200 millisecond window before the test starts. Okay, this is related, kind of scaled to the entirety of our recording period, which is about 20 minutes or so. You can see these electrodes are kind of blue. That shows that they're at baseline or slightly below. This is in z-score. So as the circles get larger and redder, that means that there's more high gamma at that specific electrode location. Okay, so the question starts. You can see this gray patch will move over to the beginning of the question, and I guess unsurprisingly, some of these auditory electrodes start to light up. There's responses that are sensory in nature. And then as soon as that critical word happens, at least down here in caudal inferior frontal gyrus, and more commonly known as Broca's region, you see activity that starts to bubble up here. This is premotor dynamics, so this is right before the person produces a speech in a 200 millisecond window, right before a speech onset of the person with the electrodes. And then speech production, which actually shows up again here in auditory cortex. They can hear themselves, and that's picked up at these electrodes. And then it's done. So this is pretty impressive, right? So we have something that has the temporal precision to actually capture this vocal exchange, and we can see this at the level of the cortex. I'll just play this movie one more time. So it starts, there's auditory, there's some kind of what looks like planning dynamics, although we'll explore this more carefully in the next few slides. That gets transferred over here to speech sensory motor cortex, and done. Okay, so we wanted to characterize and classify each one of our electrode responses across 8 participants and over 900 electrodes. And we could do it using a GLM. And the idea here is we see some responses that seem to be on during the question. So while I'm listening to the question, this is high gamma activity, and this is the same question, but actually in a slightly different syntax. So the opposite of soft is what familiar word. Soft is in the middle here. And the opposite of what familiar word is soft, we kind of put it at the end. And that exposes, I think, something interesting about these planning responses. So they're initiated by that critical word, whether they're here in the middle or they're here at the end. And then, of course, production is production. And that makes some sense. OK, so across all of our trials for these 3 example electrodes, we see something that looks like this. So each line is one trial. The ticks on the left here are the beginning and the end, in white, of the experimenter's speech. So this is an electrode that we characterized as a perception electrode. It seems pretty obvious. This is in a kind of z-score of the response at that electrode. And you can see when that experimenter speaks, then this electrode lights up. We can also align to the critical information that I introduced before and see that this planning electrode is really isolating that critical response, right? So this planning response that shows up with these ticks, which is the initiation of planning, and then those ticks that follow represent the response of the patient volunteer. And you can see that here in this motor electrode. Okay, and electrodes seem to fit into one of these 3 categories. Some had mixed selectivity, but for the most part, they fall here. And we wanted to now reconstruct in the patient's brains. So this is an amalgam of 8 different participants. And it's very subtle here, but there are little white dots that tell us where all of the electrodes were reconstructed onto this kind of canonical brain. And what we find is that sensory responses, these light green dots, are roughly where they should be. There's a small island out here that's been described by others. The motor responses are here in the primary motor and sensory, somatosensory cortex. And planning really hadn't been explored in this way, so many people had proposed that this could be in several different places in the brain, and we really see things that seem to be centralized here. If we blow that up and plot things a slightly different way and say, what is the percentage of active electrodes, it brings out what we're referring to here as an interactive language hub. So these 2 hotspots, this one is in Broca's region, so caudal IFG, and this one's in middle frontal gyrus, which is an area that really hadn't been implicated at all in speech planning. So this is something that was a kind of discovery as part of this process. So we wanted to characterize these responses, and I guess, our simple-minded idea. Is this planning for everything? Are these generic premotor responses? Or is there something kind of specific to language here? So we decided to engage the participants in a series of tasks that become more and more linguistic in nature as they progress. So the first one is just generic premotor. They have a finger button, a kind of spring-loaded button that they can hit a variable number of times. And we really don't see this area of the brain being engaged with that task. There's orofacial non-speech, and now we're starting to engage the muscles that are involved in speaking, but in things like puckering your lips, sticking out your tongue, smiling for the camera, all of these things, and we don't see much. We start to really engage the network with speech tasks, and in a very simple speech task, which is just handing back a word that's given to the participant. For example, say ba clearly into the microphone. Those repetition tasks start to engage portions of this, and then it becomes fully engaged when things like lexical access happen, and we have to have the person go out and find the plural form of a word, in this case, goose, clearly into microphone. So as these tasks become more and more linguistically relevant, then it starts to engage this network more and more. So it seems like the planning network is preferentially active during linguistic tasks. So that's fine. I've told you we've used the tool that was developed by Sarah Bogles and Levinson, this kind of critical information task, to get to planning dynamics, to isolate those dynamics. But we wanted to know if this has anything to do with conversation, because we're not sitting here, engaging with our friends and saying the animals who moo say what or whatever. And so what about natural conversation? And so we can actually engage these participants in natural conversation with these electrodes still on the brains, on the cortical surface. And we can see what we see during these kind of totally unstructured interactions. And I think what comes out is pretty magical. So this is example high gamma responses from two different electrodes. Right? One is in caudal IFG, and the other one is here in a motor region. And this is just part of a conversation, right? So the experimenter is shown in gray, and the participant is in white. Were you born in Mexico? New Mexico. Have you been to Mexico? No. Never? Never. But when that person talks, you can see this electrode, which is in a motor region, has a clear motor response. But what's kind of surprising is when we look at Broca's region, it's not active during this kind of spoken task. It's really active more in this planning side. So there's an alternation of what's going on in Broca's region and what's going on in these motor centers during this totally unstructured speech. So we wanted to take a look at the population of responses. These are all of the task-engaged electrodes for one participant. And we wanted to look at them in a more unbiased way. So we just did a principal component analysis on all of the activity during this task epoch. And when we did that, we saw that the activity did cluster into these three clusters. And we wanted to find out if the same clustering could be observed in conversation as well. You can imagine maybe it does. That's just what the brain does. I don't expect that auditory cortex is going to move around that much, for example. But it could well be that conversation is a different state, the person's in a different state, and things get mixed up a little bit. And what we see, indeed, is support for our model one, where things really are, they remain clustered. The activity during conversation really does mirror that, what's going on in the task. And we can paint each one of these dots with the designations that we made in the GLM analysis I told you about before, and see that the roles of each one of these dots remains the same in the two different states. And we could take the consensus activity of each one of these clusters and reconstruct a kind of time-varying principal component. And if we do, we see that the dynamics are really similar across the two states as well. And so this kind of thicker line here represents the average perception PC, and the blue line represents the average planning PC. And you can see both for critical information task as well as conversation, things are active here during the experimenter speech. That's not entirely surprising, but the planning happens after that, when the person is getting ready to say what they have to say. And there's also the same kind of thing observed here. If we align to participant onset, planning precedes production in both cases. So we find that these identified networks behave similarly during interactive tasks as well as natural language use. So we can't do a lot of the same things in this setting with these patient volunteers that have given their time to us for these studies to really piece apart a circuit or to ask any questions about necessity and do they look like planning? I noticed there's something in the workshop, everything is everywhere, so it could be planning is just something that's broadcast across the brain. We could start to get a little bit at causality with experiments like this. So in 4 patients, we've administered a small amount of electrical current, and we can do that in sites that we designate to be planning sites using the kind of analysis I've already walked you through. And if we administer that small amount of current in half of the trials, we can start to see if we see any behavioral differences. Okay, so these are, it's a critical information question. It's schematized with the question shown in gray and the critical information in blue. And this is a bank of questions and responses that are control. We're not stimulating at all in these cases. And here we are, and the stimulation is shown with a yellow line. So it's administered kind of haphazardly. We're trying to control now where that stimulation is given. But now it's given in half the trials, okay? And we want to know if that affects the fast back and forth that we see under normal circumstances. So this is the turn-taking rate of this specific participant. And when we stimulate, we see that that rate slides over and increases by about 50%. So we've slowed down these fast interactions. But what I think is really fundamental and really amazing, and we're still trying to get our head around this, is some of the errors that arise. So the person can still speak. They don't go into a state of speech arrest. So if the same kind of current is administered to motor regions, for example, then they'll have the experience that their tongue is frozen and they can't actually make words. But here they can speak just fine. But what seems to be really affected is planning. So this is a question, say the plural of man, clearly into the microphone. The stimulation is given in yellow, and the answer is man. Well, the correct answer is men, but here it was returned as instead of doing a pluralization task, they've almost reduced this to a repetition task. Which word is the opposite of fast? Well, during stimulation, this participant said rapid. And then when the stimulation ended, said the opposite would be slow then. So able to correct themselves. And that's something that's, I think, really interesting. We're trying to put our head around this, and we've seen very similar things across other participants. And in other participants, we're able to stimulate other areas and see no measurable effect as well, including areas that are quite close to this region. So it seems to be something that's really selective for specific areas of the cortex. So to take you through a summary of this first and most kind of significant part of the talk, speech perception, planning, and production activity are separately represented in the human neocortex. A language planning network necessary for interactive speech includes the middle frontal gyrus and Broca's region. The interactive planning network is primarily tuned to linguistic operations rather than generic motor planning. Consistent cortical responses occur during scripted tasks as well as natural conversation. And finally, perturbation of activity in the language planning network leads to slowed interactions and linguistic errors. So if we go back to this kind of schematic, this framework, we looked at this as maybe an opportunity as well. So when I look at this as a systems neuroscientist, I feel kind of uncomfortable because boxes and arrows make me crazy. I want to learn something about what's going on inside those boxes. This is what I've been doing for most of my career. And we'd love to model language, but there is no good animal model for language. Language is something that is really specific to humans. So I think what is good about this is we've kind of fanned this out. We've been able to take something like interaction and spell it out in a number of different modules. And maybe animals are one way of addressing specific parts of this. So, for example, there have been a number of labs that have made progress on how mus musculus make their vocalizations, and this is a sonogram of that. So frequency is here on the y-axis, and time unfolds on the x-axis. And that really, I think, is applicable in this case to the affect pathway, so state processing and emotional readout can be looked at in the mouse. So all of the tools that have been developed in the mouse can be used to do some very elegant work that's been published quite recently. So the content pathway, there are a number of creatures that can be used to examine that, and this is, I think, a particularly striking one. So in the vervet monkey, they make these warning calls that they've learned to associate with specific environmental stimuli, including, for example, a leopard. In this case, there's a predator coming and can unveil this kind of warning call. The songbird is an animal that I've worked with for some time now, and I think we can address certain parts of this, right? Certain modules, and I want to say that I'm particularly excited that Yarden is speaking after me. He's doing some very exciting things with canaries. And just to give you a feel for the kinds of things we can do to start looking inside these boxes, we've primarily focused on the motor processing end, and we've been able to use a lot of different tools to get to how that works in this creature. And then maybe from this new framework, we can take the results that we found and put that into a larger kind of discovery. I promised Jürgen I'd show this movie, even though it increased the size of my presentation by a factor of 5, but it's always good to see. And the last part of this talk, I want to tell you about a new model system that we've begun to explore to get to some of these boxes, including the boxes we think are going to be critical for interactive speech. And that's the singing mouse, or Scutenemus teguina. And this is a group effort. So Daniel Akobe, an former MD, PhD student in the lab, really started this. Steve Phelps has been characterizing these mice in a different context, a kind of Evo-Devo thing at University of Texas for many years. And Daniel wanted to put them into the context of systems neuroscience. And this has been helped out significantly by the efforts of Arko Banerjee and Yuki Fujishima, with some computational support by Druckmann and his very talented student, Feng Chen. So this is what that looks like. These are two animals. They're in different terraria, and they're interacting with each other vocally. So this first animal is going to make what's referred to as an advertisement call. And this animal will pay attention to that call, kind of spin around, and then launch a response as a result. I'm always curious, who could hear the mice sing? Okay, most people. You're all so young, this is great. So the song structure looks like this. The song is typically 5 to 12 seconds, give or take. And it's made of these down sweeps that become longer and more elaborate as the animal sings, and then kind of loses some steam at the end. I've plotted this thing, this trajectory where I put a dot at each one of the notes, and the vertical position of that dot tells us the duration of that note. So you can see this animal is singing and produces this long advertisement call. And does so in an interactive way. So as you see from the movie, many of you probably think of, I don't know, extracellular units, and these are two channels, and they're very correlated. And, but these are actually, it's about 2 hours of singing activity from these two animals. And each vertical tick is an episode of song. So this white animal will sing, and the red animal will respond. This is a kind of triad with red, then white, and back to red. And we can look at the timing of that and align to the end of the white animal's song, as we do here across many trials, and see that the red animal picks up with a really minimal latency, in this case about 500 milliseconds. So if we compare that to human speech, we see that it qualitatively matches those dynamics. If we look at marmoset, and so this is a creature that's really been the most studied in terms of interactive vocalizations, it's considerably longer. People who have looked at this and done some really beautiful work in this realm suggest that maybe this is driven by a different circuit. For example, something that is more emotional, cingulate cortex arousal kind of circuits, and if one stimulates the cingulate cortex, it causes an increase in heart rate, and seconds later, a phi call like this. So it seems to be a different type of behavior. So we wanted to find out if the neocortex was involved in this. And the way that Daniel did that is he stimulated in a patterned way across the neocortex and recorded from vocal musculature and found this hot spot where a small amount of stimulation resulted in the flexion of those muscles. And Arko Banerjee then put electrodes into that area of the brain, into the orofacial motor cortex, And this is what's going on across 28 neurons that are roughly in layer 5 of orofacial motor cortex while the animal is producing his advertisement call. Spikes are happening. And we can take each line here and plot it in a kind of grayscale plot where the spike rate is shown here. 20 hertz is white, and down here is 0. And you can see across these trials from the beginning of song, shown with this kind of dashed green line, to the end of song, which is with the red line, that there seems to be a repeated cortical dynamics as the animal sings over and over. And if we look outside of song, these are just the same duration, but taken in some episode outside of the context of singing considerably less structure. And we could zoom in on some of these neurons and see some really intriguing stories that are told. So this is social modulation at the level of a single neuron. That was actually one of the ones that I showed you here. And so this is a raster plot, and the song duration is shown with this kind of grayed area here. And I can also plot it with these black ticks and see that there's activity here that happens while the animal is singing. But there seems to be some heterogeneity before the onset of song. Sometimes there's a lot of firing, sometimes considerably less. And so we could break this into two different categories, look at the animal sing in a kind of alone condition where he's singing by himself, and this is in response to a partner. And you can see the partner song is shown with the beginning and the end, like this. And things really heat up as that partner sings, and this is probably something to do with motor preparation that could be similar, at least in flavor, to some of the themes that we touched on in the human recordings. So how can we really figure out what the motor cortex is doing here? And so, you can imagine there's a group of cells. I don't have to tell you guys about manifolds, but this group of cells could have some series of solutions as the animal is singing that leads to behavior. So this is our kind of song, if you like. And we wanted to explore this, but we found that when we stimulated anywhere in the brain, it just kind of stopped the behavior. And this makes some sense, right? So if you knock out this activity, then it just goes off here, and the behavior itself is compromised as well as the population activity. So we decided to turn to an old friend, which is focal cooling. And the idea there is we could actually focally cool different parts of the brain. Here we started with the orofacial motor cortex. And things can still travel along this manifold, but they do so with the slower time scale, right? So this is stretched out in time. The behavior is stretched out in time. And we can see if we can learn something from this approach. So there are 3 models of what we might see here. So one model is if we cool orofacial motor cortex, if everything is in there, the notes, the song, all of these time scales, we'd expect what we'd seen many years ago in the songbird to occur, right? So the song itself would just kind of uniformly stretch out, and that would look kind of like this, right? The notes and the song would just swell. Well, it could well be that everything is subcortical, right? So the house mouse's song is profoundly subcortical, and in fact, this is quite an amazing example of animals that are born without a forebrain at all, and they make normal song, at least a statistically indistinguishable song. So it could well be we cool down orofacial motor cortex, and we see no difference at all. Or it could be that this thing is somehow hierarchically organized, that certain things are coded up in the cortex, and certain things are coded down here in subcortical areas. We got a hint of that when we started looking at the songs in different contexts. So when the animal sings by himself, then the song looks really stereotyped, and this is again that song trajectory plot that I showed you before. But when the animal is singing in response to some vocal partner, it mixes it up. It can stop and start, it can bend the song, break the song. And so we think that maybe this is what cortex might be doing, right? Adding this kind of flavor. But interestingly, and I don't really show it clearly here, but we have in a different analysis, the actual maximum syllable length doesn't change. The minimum syllable length doesn't change. When songs are longer, they just simply add more notes to get from point A to point B. So that suggests that the note generator could be elsewhere. And so maybe cooling OMC does this. It can change one time scale, which is the song duration, but without changing the actual notes that are produced. And in fact, that's exactly what we see. So when we cool, the song itself stretches out, but this thing doesn't tip up. We don't actually bring in longer notes suggesting that the motor cortex is creating the notes. We think that this kind of thing is absolutely perfect for giving us the countersinging that we've seen in these animals. right? So this is going to be executive control, song modulation. This could be more note production, and maybe this could lead to countersinging. So we did this one final experiment, which is to have the animal duet with a loudspeaker. So we could play sounds through this loudspeaker. They respond to the loudspeaker. We have more experimental control that way. There are vertical ticks here. The vertical ticks are when the loudspeaker makes the conspecific song. The circles here are the responses of that mouse to the conspecific. And you can see there's a very faithful countersinging to the speaker. If we inactivate OMC with mucimol, that faithfulness goes away, right? It's just kind of reduced to a few examples. But I'm really glad the examples are there, because we look at the time scale and now we see that this is something very different, right? All of the fast responses are abolished, and we're left with just a few very slow responses that really have the time scale of the marmoset, if you like. So we think that this could very well be inputs from cingulate or maybe these other arousal places that are triggering these songs. We haven't done that experiment yet, but we're excited to do that to follow up. So as a final summary slide, in both the cases of human speech and the singing mouse, there's some kind of a sensory trigger. We've used critical information as well as just open conversation, where different triggers turn in prediction is something that's in kind of discourse analysis, things that can trigger that person to respond from a partner in this incredibly efficient, incredibly fast way. And here in a partner song, we're interested in specific features that could do that. There's going to be some kind of motor preparation and interactive language hub in humans, and here orofacial motor cortex, and that leads to fast spoken exchanges and countersinging. We can perturb this by focal stim disruption in human patient volunteers that then can lead to slowed interactions or linguistic errors, pharmacological inactivation with the GABA agonist mucimol that then abolishes countersinging. And if you like the kinds of themes that I'm made an attempt to bring up today, then please look into this new Gordon conference. So Sarah Woolley and I have put this together. It was kind of shuttered by COVID in 2020, but we're making a run at it in July, early August in Mount Holyoke College. Registration has started, and it's filling up pretty quick, but if you're interested, please do register for this. It'll be bringing together some amazing people who look in the human brain, Eddie Chang and some other people, as well as a whole variety of creatures, from singing fish to naked mole rats, and how they do what they do. Thank you.
If you have questions, please go ahead.
Hi, Michael. Thank you for that beautiful talk. I especially appreciated the diversity of animals that you managed to feature in one talk. I'm particularly fascinated by the little piece of preliminary data you showed about naturalistic conversation. So I noticed that in that example, it was still very much the experimenter talking and then the participant responding to a question. I'm curious if you have thoughts about what happens if this were an even more naturalistic conversation where I'm expected to actually come up with the next topic in addition to simply responding to what you're saying. If you thought about that and what the neurocorrelates that might look like.
It's a great question. So we have a lot of conversational, hours of conversation really to mine on this. And so the one piece that I had was very kind of question and answer, but it flows very naturally into something that is less that way. Mm-hmm. And we're working with Jelena Krivokopik, who's at the University of Michigan, who is an expert in discourse analysis, to try and piece this apart. I mean, obviously, people have talked about structured laboratory tasks versus things that are more natural. The beauty about structured laboratory tasks is it makes analysis much more straightforward. This is something that is going to be hard, but with Jelena and other people's help, we hope to get to the heart of it. Yeah, it's a great question. Thank you.
Thank you. I look forward to hearing more about it.
Let's take one more from this side, and then over there.
As an Israeli, I'm compelled to ask whether there's preparatory activity that's different when humans or animals interrupt their...
Haven't done that. It's a great question. Great question.
Hey, super cool and super imaginative use of everything, different species, different, wonderful.
Thank you.
so strikingly different things between species in the brain anatomy, right? So motor cortex in a human will control the actual execution of the movement, but it seems like in the singing mouse, you're saying it won't, and motor cortex is playing the role that premotor cortex or planning cortex is playing in the human. Is that what you're saying? And if so, maybe you can comment on that, maybe with reference to what happens in just simple planning of actions, differences between humans and mice as well.
Yeah, I mean, look, I think going back to Mar, I mean, there's implementation that's going to be different across species. I'm more interested in the algorithmic level and what's going on. So sure, there's going to be differences in the mouse versus humans.
But is that true that effectively, I think you're saying that motor cortex is actually important for planning the action in the human, but not in the mouse. That's what you're saying, right?
So we've done motor cortical lesions in the mouse, and they're still able to sing, maybe driven by some of these more emotional centers I've referred to before. Yep. Thank you.
Mike.
Oh.
Fantastic, as always. So I was struck by something that I hadn't noticed before. You know this much better than I do, and probably anybody else in the room. In the zebrafinch, the males have this perfectly crystalline song when they're directing their song, and exhibit much more variability when they're by themselves.
Right.
What you showed with the mouse was exactly the opposite. I sort of understand, or I think I understand, what the bird is doing. Why would the mouse exhibit so much variability?
Yeah, that's a fantastic question. So people like Alison Dope and others have started looking at things like directed versus undirected song. Undirected song is when the zebrafinch sings by itself, and as you said, has much more variability. Vikram Gadekar and others have also explored this, I think, to really nice effect. That's more, I think, about reinforcement learning and the idea that adding in that injection of noise when the animal is practicing can help to just sharpen the song. I think the variability here is kind of more pedestrian. The animal is singing, and it's singing in response to a partner. So the variability is because it's weaving in and out of its song in response to that other animal, right? It doesn't, the idea of turn taking is to minimize when they're both going at once and to kind of alternate. And so that's the source of that variability.
Oh, hi. I noticed that when you inject a current to the participant, you ask what's the opposite of the fast, and he says rapid. So did you collect, is there a pattern when they answered wrong? Because it seems like the correct answer supposed to be hijacking some sort of default pathway that seems to be a synonym of the correct answer. Did you look into the wrong answers?
Yeah, so the pattern often is that they kind of devolve into repetition as opposed to making some kind of computation on the word, right? So if you look both in the case of man and men, they did not pluralize that, right? Or in the opposite case of fast and rapid, they didn't make an opposite there. So that has to do with actually accessing the right word and then putting that into some kind of articulatory coordinates. But yeah, that seems to be the pattern, and we've done this now on 4 participants, and we're still actually actively analyzing the data, so stay tuned.
Okay, thank you.
Thank you.
One last question. Thanks for a great talk, really enjoyed it.
Thank you.
A lot of things what you did was like modifying what they might be doing. And I suppose in this context, at a conference that serves beer and wine at the poster station, how would alcohol affect response planning? Especially when maybe it would be, I'm planning to respond to what I said previously, not what you would be saying.
How does alcohol affect interactive speech? Ask me at the poster session. Thank you, everybody.
Thanks.
Let's thank Mike again. All right, our next speaker is Yarden Cohen from Weizmann Institute. He's going to tell us about a new tool for automating annotation of complex birdsong, reveals dynamics of canary syntax rules.
No, yeah, okay. I'll fix it. I don't need this. Thank you. Okay. Good luck to you. Thanks.
Okay. So, okay. Thank you so much. My name is Yarden, and today I hope to show you how we can automate the annotation of complex birdsong, and then how we can use this tool to ask new questions about syntax and its neural encoding. So many of our behaviors are sequences of motor or vocal elements. In some cases, some behaviors, the steps are clear, but even in seemingly continuous behaviors, they can be composed or thought as sequences of more basic elements. And the order of elements matters. For example, this dancer may produce or change the sequences of her dance moves, and some sequences perhaps will be of greater beauty. And this relation between syntax and semantics is, of course, most common in language, where the sequence of words determine the meaning of a sentence. So we can, and specifically at certain time points, a transition to the next word will depend on several previous elements. So we can probably say canaries like to, and then sing or fly, maybe with equal probability. But if I replace the word canaries with, let's say, neuroscientists, then now I'm most likely to say neuroscientists like to sing, not neuroscientists like to fly, because, well, I'm sure some singers in this audience know I'm referring to them with this example. And so you're awake, right? OK, so this point here, this transition here, that depends on several steps back, is a high-order syntax rule. And I will refer to those as complex transitions in this talk. And in my lab, we study how the brain learns, controls, and perceives the syntax of flexible behaviors. And we do that in songbirds, exactly because they produce these sequences of well-identified syllables, which you can see. If you actually look at them singing in the morning, when the air is cold, then you can observe every syllable and every breath coming out of their beaks. Another advantage of songbirds as a model is that they sing a lot. And for the past 20 years or so, studies in songbirds really benefited from automated methods that allowed scaling up experiments and leveraging these large data sets. So for example, a method for measuring song similarity really benefited and advanced studies in zebra finches because these species learn to produce a very repeating song motif, for example. But there are other questions we might want to ask that require that we annotate every syllable. So for example, the Bengalese finch will produce songs that sound sometimes like this. But containing more repeats and maybe more, and also more diverse sequences. And to annotate those songs, we use the sonogram, so the time and frequency representation of audio, and also the amplitude, which we then use to find segments of threshold crossing. And then a human annotator comes and assigns a label to each one of those segments. And this is the time-consuming step that actually limits the questions that we can answer. And so in the next slide, I'm going to show you how we can sometimes automate this labeling. But even before that, at the segmentation step, we already introduced a lot of error. So for example, we'll take from a canary. Canaries produce syllable repeats, that are called phrases. And let's say we want to segment these syllables B here. We cannot do it without breaking those syllables A or joining those syllables C. I'm sorry that you probably cannot see it. But in any way, this is not even a full canary song. This is just a short example of a canary song is much, much longer and contains hundreds of syllables. So let's play one, for example. Good. Now you're laughing. This was one canary song, okay? And I can play you one after one, song after song of the same bird, and they never repeat. Each individual canary chooses from a repertoire of between 30 and 40 distinct syllables to compose their songs. They always produce this hierarchy of syllables in phrases, and they can flexibly control the sequence of those phrases. They also change their song between one year and the next, and they undergo massive neurogenesis in this process. So this, perhaps, is the reason why no prior studies really automated the annotation of canary song. So what happens if we naively try to train a machine learning tool, let's say a support vector machine, to label those threshold segmented birdsong? We can do it using a variety of training set durations, and we can measure the syllable error rate, which is the fraction of edits we need to make to actually correct the test set predictions to the ground truth. And it doesn't work really well. And the reason it doesn't work very well is because of those errors introduced in segmentations. In fact, if we actually manually proofread the segmentation, we get a dramatic improvement. But this is exactly the labor that we're trying to avoid. So what we did is to develop a deep learning algorithm that we call Twitternet. It actually fuses together these segmentation and labeling steps and leverages the training data to reach a performance that is almost as good as the ideal classifier. And it asymptoted about 0.9% error at Bengalese finches and about 3.1 in canaries. And so how does it work? Well, Twitternet acts on spectrograms, and its output is the likelihood of each one of the syllable types and a background class in every time bin of those spectrograms. To learn to do that, Twitternet learns localized features in convolutional blocks and temporal dependencies in a recurrent layer, and then a projection onto the different syllable types and the background class. Now, this is just recently published, so the design and its considerations, as well as how we actually tune all the hyperparameters, is all there, and you can go check it out. But to go from these metrics here to the output, to the segments we want, we first assign the most likely label to every time bin. This already introduces errors. For example, spurious vocalizations that you cannot see there are getting labeled, and syllables that are very much alike will get confused. And so what we do is we apply two post-processing steps. One is to reject very short segments, and the other one is to take every background separated segment and assign one label to all time bins using a majority vote. This turned out to actually increase performance by a lot. And when we asked why, we found that just by correcting very few time bins that are erroneous, you'll avoid these extra segments and you'll avoid splitting longer segments. And so, as I said, Twitternet reaches about 1% error in Bengalese finches and about 3 in canaries. Is it good enough to replicate past results? Well, yes, and here's a few examples. The Brainerd lab, or past studies in the Brainerd lab, showed that Bengalese finches have a stable or maintain the transition probabilities across a month or two, and they did it by measuring the probabilities of transitioning between, let's say, syllable E to F or I over a few days, and then repeat it after a month or two. And so what we did is to take about 10 minutes of data and train a Twitternet model and predict data for about 3 or 4 days, and see again the same stability of transition probabilities, as well as if we look at all the ground truth transition probabilities and compare to the model prediction, we get almost the same results, or hardly distinct. In canaries, the sequences of phrases that you see here annotated in color and also in letters are flexible, they're variable. And past work in the Gardner lab showed that actually following certain phrase types, there's a transition that depends on a long-range song history. It can be seen here in an example. So following this pink phrase, there will be several outcomes, and this is the likelihood of those outcomes. It will maybe be the same if you condition on one step back in the sequence of phrases, but maybe changing dramatically if you go 2 steps back. And so, again, this transition here is exactly that same kind of complex transition that we saw in word sequences. And work by Jeff Markowitz showed that you can capture these kind of syntactic dependencies as a branch in a model called the probabilistic suffix tree, basically describing all the Markov chains that impact this transition. And so to replicate this finding in a data set that is 3 times larger, for example, we first found all the branches in one canary that describe all the syntax rules or dependencies of transitions following all phrase types in this one bird. And then we took about 19% of the data, and we trained a Twitternet model and predicted the rest of the data, and got almost the same results. Now, this branch here is missing, exactly because it's very rare. And we found an almost linear relation between the number of songs we can use and the probability, or the minimal probability of an event we can include in these models. And so now that we have the ability to basically annotate many more songs, what can we do? We can start looking at, say, the neural encoding of canary syntax rules. To do that, we implanted a miniscope on top of HVC in a singing canary, and here's how the data looks and sounds like. What you'll see here on top will be the video, what this camera here sees. It will be background subtracted. There will be annotated regions of interest. That will be all the cells that were active during that day. Below that, you'll see the raster of activity of these neurons. And at the bottom, you'll see the amplitude of song, the song amplitude, which will be color-coded for the different syllable types. Okay, so when we looked into this data, we found cells that are context sensitive. For example, this cell here, it's active when the yellow phrase appears here. It's not active when the same yellow phrase appears in a different time point. And there's many cells like it in this very film, some that are active strongly in one context and weakly or not at all when the same phrase appears in a different context. And it turns out that this context sensitivity relates to the sequence of phrases. So let's look just at an example. Here are two cells that are active during this orange phrase. And each of them is active preferentially following one of these contexts. You can see it if we color code the context sensitivity of every fluorescence video frame, but also if you look at the rasters of activity, so here's that, the signal is time aligned to the onset of the orange phrase, and rows or songs are separated by the different preceding contexts. And during that day, in a sequence of 4 consecutive phrases, we found cells that carry this sequence information. It bridges across about 4 seconds of behavior and about 40 syllables. And when we ask in which phrase types do these sequence-preferring cells mostly fire, we found that those are exactly the phrase types that participate in complex transitions. So if we stand on the shoulders of studies in zebra finches, this activity of these neurons marks the activity of neural states, or a precise motor program that underlies the production of syllables. And past studies also show that multiple steps can actually map into the same syllable type. So what these findings suggest is that canaries may be using those redundant representations as a short-term memory to carry the syntax information that they require for their complex transitions. And so finally, that we have now annotated data sets that are orders of magnitude larger than previously accessible, we can start looking into the dynamic or stability of canary syntax rules, or canary syntax. So for example, we can look at the probability of transitioning between phrases. And in some cases, those are stable, again, across days, as I showed you on Bengalese finches, but in some cases, they're not. So every dot here marks a point that is significantly different than the first day. And if we look at all the transitions of one bird, we'll see that many of them change across weeks, basically. And then we can also look at transition points, meaning all the outcomes from every syllable, from every phrase type, and ask which one of these transition points actually changed significantly across week, and whether it's increased or decreased its entropy, or became more or less random. And here across 4 birds, there is no consistent trend. However, we then look at second-order transitions. So transitions from this phrase B, but here I'm separating in these subplots the different histories of B. And again, very different dynamics in very different contexts. And when you look at the second-order transition probability, transition points, basically, we find that now there is a consistent finding. In all 4 birds, more transition points became less random or decreased their entropy across weeks. And this suggests an emergence of a sort of a long-range order. So just to show you one example, the bottom here is the probabilistic suffix tree of one bird in the first week of recording, and the top one is in the third, where you can see greater, just more, and deeper dependencies on the history of song. So with this, I'd like to say many thanks to many people in the Gardner Lab that's contributed to this project, and a special thank you to David Nicholson, the co-developer of TweeterNet and a real gentleman. And lastly, please reach out to us if you want to apply TweeterNet to your data. We're running tutorials these days, and we're already working with about 10 labs. My lab just started about 6 months ago at the Weizmann Institute, and we're hiring, so reach out to me if you care about the building blocks of neural syntax, if you want to study syntax learning, maintenance, or perception, and if you care about functional circuit dynamics or neurogenesis as well. Thank you so much for your attention.
Okay. We have time for a couple of questions.
Can't see.
Maybe I could ask a quick question. Is TweeterNet going to be on Twitter soon?
Can we teach it how to tweet? Yeah.
No, that's a serious question. So in your architecture for TweeterNet, you had LSTMs, and then you had a correction afterwards with the majority voting.
Yes.
Is there a way to eliminate that majority voting step in your architecture of TweeterNet?
I can tell you what is our next step. Is to include in the loss function also a term for the actual syllable errors, and not because right now the error is just on the frame error rate or the bean-wise error rate. It doesn't penalize for breaking syllables. So if you just add one error of course, you can correct for that too, but of course, if you add this one erroneous frame, you break a syllable, and you really increase the syllable error rate. If you penalize for that, then this is the way I think we should go.
Thank you. We have a question there.
Hi, maybe here.
Can't see anything. Hey.
Hi.
How are you?
So on your preliminary data, you show, in a way, an increase in stereotypy of the two transition syntax, right?
Yes.
You think this is kind of the seasonal process going on, like the song being more stereotyped because you approach spring or summer, or do you think it's a different? So the time here is aligned with the seasonal process, right?
Right. I mean, time is, first of all, aligned with time, right? Yes. Whatever's kicked in, whether it was placed in the experiment, even if you wait a bit, I mean, there's always time. So, I mean, I'd like to hope it's the case,
so it's a critical,
but it's preliminary.
It's a critical time in the season, or?
This was all recorded in the middle of spring, basically.
Okay, and so they were all at the same timing during spring?
Yes, they're all in the same timing, the same experiment, yes.
Thanks.
You're welcome.
All right. For the time, we'll start the questions here. Okay. Let's thank Yarden again. Okay, we have another contributed talk. This is by Sandra Romero Pinto. She's going to talk about linking tonic dopamine and bias value predictions in a biologically inspired reinforcement learning model.
Do you hear me well? Yeah, okay. Cool. Thanks to all for coming today. I'm very excited to talk to you about my current project regarding how to link tonic dopamine with biases in value predictions using reinforcement learning models. Okay, so let's get started. So a certain hallmark of psychiatric disorders is the presence of abnormal value predictions that can lie within extremes of this spectrum. On the one hand, patients with major depressive disorder will present decreased learning from gains and pessimistic expectations. And on the other hand, like patients with addiction, pathologic gambling, and mania present optimistic expectations and enhanced learning from gains. And in this talk, I will try to tell you how, by using RL models, we can link these two axes of behavioral symptoms, the asymmetry in learning with the biases in value predictions, and answer the question of how can reinforcement learning models give rise to these biases. And I will point out to the effect of learning rate asymmetry in the emergence of these biases. And in addition, and most importantly, I will try to link this axis of asymmetry in learning with dopamine, and basically try to answer the question of how can dopamine be implicated in the emergence of these biases. So, to understand this talk, we just need to know a bit about RL. So basically, in RL, there's an agent that's trying to maximize its rewards. And in order to do this, it will learn a value function that is defined as the expected sum of returns held by different states. And learning of this value function is driven by reward prediction errors. And these RPs, or reward prediction errors, will be used to update the value estimate with a certain learning rate that I will call alpha in this talk. And basically, with this very simple learning rule, the value prediction will always converge to the expected value of the reward distribution. And so, for example, here, if we have a distribution that is Bernoulli distributed with probability of reward of 0.5, the expected value is 0.5, and the learned value will converge to that estimate. And now, if I have different reward probabilities with the same type of distribution, the learned value will be scaling linearly with the probability of rewards. And so now, how can these reinforcement learning models could give rise to biases? Basically, as I said, this can all be rooted in the learning rate asymmetries. And so we can now actually allow an agent to perform the value updates with asymmetric learning rates, so alpha plus and alpha minus here for positive and negative RPs. And if these learning rates are asymmetric and the agent or the animal is now encountered with the same probability of rewards, the value estimates will actually converge to estimates that are larger and lower than this value, leading to optimistic or pessimistic biases. And so basically this level of asymmetry can be summarized by this asymmetric scaling factor that I will call tau here. And so a standard reinforcement learning model will be the special case of a tau equal to 0.5 with equal learning rates for positive and negative RPs. But now if this value of tau is like larger than 0.5, for example, the alpha plus will be larger than alpha minus, and this should give rise to optimistic biases. So here, for example, for this reward probability of 0.5 that has an expected value of 0.5, the learn value will be larger than that. And the contrary will happen for values of tau lower than 0.5. And so this is something that was developed before and is what we call a risk-sensitive RL. And basically, in the risk-sensitive RL, the biases in value learning can be summarized by the star parameter that will modulate the level of the bias. So this asymmetry in learning rates is actually a key feature that stands out when you feed RL models to behavioral data from patients with depression, for example. Here, anhedonic symptoms in depression correlated negatively with the learning rates from rewards. And in this study, learning rate from losses was not affected. So this will actually give rise to asymmetric learning rates and lead to pessimistic expectations, as the ones that we usually observe in patients with depression. And in the study as well, what they saw is that after therapy, the improvement in symptoms was actually correlated positively with the improvement in learning rates from rewards. So basically taking like the learning rate asymmetry more to the right of the spectrum. And so with this in mind now, we can actually equate this spectrum of biases in value predictions with this tau parameter that's like telling us how much asymmetry there is in our learning rates. And so now we'll just like try to understand like what rate mechanisms can actually give rise to these biases, or to these actually asymmetric learning rates. And so I will I will tell you about two models. One is the one that I'm proposing. The other one is like something that could also be happening. So our proposal is mainly based on like the function of the baseline dopamine in development asymmetric learning rates, and I will call model 1 this one. And in addition, there's like another proposal in which like the asymmetries in learning rates could be based on phasic dopamine responses, and I will call this model 2. Okay, but for looking at mechanisms, we need to know a bit about how RL happens in the brain. And it's basically in the brain, dopamine ions are thought to encode for reward prediction errors. And something that we need to take into account now is that these dopaminergic RPs will modulate plasticity in downstream targets. And among these targets, we have the spiny projection ions in the striatum that are thought to represent for value. And basically, the idea is that dopaminergic RPs will generate plasticity changes that will update these value estimates. Something that now we need to take into account is that actually the dopamine-dependent plasticity is a function of what receptor type the SPNs will have. And so recent evidence has fortified this idea that dopamine bursts or positive RPs will generate LTP in D1 SPNs, and dopamine pauses or negative RPs will generate LTP in D2 SPNs, LTP being long-term potentiation. And so with these plasticity rules in mind, we can now implement a narrow model, but with separate populations of value predictors that we'll call P and N. And mimicking the effects of dopamine plasticity, basically the P cells will increase their estimates if an RP is positive, and the N cells will increase their estimates if an RP is negative. And just in order to keep this model working as standard R models, the final value predictions is the difference between the P and the N estimates. Something also that we can keep in mind here is that now we have two separate populations of value predictors with separate learning rates. So this model kind of allows us to just scale separately the learning rates for each of them. And so now if we can think of a mechanism that can generate this asymmetry, then we can actually reach regimes of risk-sensitive RL or biases in value predictions. And so in order to understand that mechanism, we can now take into account another biological feature, that is that dopamine receptors will have different affinity to dopamine. And these different affinities of dopamine can actually be better observed if we look at the receptors' dose occupancy curves. So basically, this curve is describing how much the receptor is occupied depending on what concentration of dopamine there is. And because of their different sensitivities, D1 and D2 receptors have similar shapes of this curve, but they're shifted between each other with respect to baseline dopamine. Basically, D2 receptors have a higher affinity to dopamine than D1 receptors. And so basically, if we assume a baseline dopamine of 60 nanometers that has been reported, starting from this baseline dopamine, a pass on a burst can cause similar changes in receptor occupancy in either of the receptors. And this can result in similar receptor sensitivities. But now if we actually decrease the baseline dopamine level, it will hit a different part of the sigmoidal dose occupancy curve, and this will cause a greater change in receptor occupancy for a burst than for a pause, so basically making D2 receptors more sensitive than D1 receptors. And the contrary will happen for high levels of dopamine, where now D1 receptors are more sensitive than D2 receptors. And so we can do this same exercise for a sort of baseline dopamine levels, now here represented with a color map, and we can reach this relationship where now receptor sensitivity for D1 and D2 receptors are modulated by the baseline dopamine level. So now, in our model of PNN populations, we can assume that these learning rates, alpha plus and alpha minus, are a direct function of the receptor sensitivities. And something interesting here to keep in mind is that this effect of the shifts in baseline dopamine on the sensitivities is opposite between the receptor types. So basically, if I have a high level of dopamine now, I will have D1 receptors that are more sensitive, leading to an asymmetry in learning rates that is in favor of the alpha plus. And the contrary will happen for decreases in dopamine, where now the asymmetry is in favor of the alpha negative. And so we can summarize now this, or reduce this asymmetry in learning rates to this parameter of tau, which is a symmetric scaling factor. But now this tau parameter is actually changing as a function of baseline dopamine. It's actually increasing with baseline dopamine. And so taken together in this model, changes in baseline dopamine can lead to asymmetric scaling of value updates, summarized by this tau parameter. And as I showed you before, just by having this asymmetry, we can reach biases in value learning. But now we can actually draw these biases as a function of baseline dopamine. So now we can define model 1, where this asymmetry in learning rates, summarized by the tau parameter, is a function of baseline dopamine, and the alpha plus and the alpha minus are proportional to the D1 and D2 receptor sensitivities. And now we'll just look briefly at the model 2. So the model 2 is, so the model 2 is based in this model, the asymmetry in learning rates, so the alpha plus and the alpha minus, are now a function of phasic dopamine responses. And this basically corresponds to the slopes of the dopamine response functions for RPs. And if these slopes are symmetric, then this mechanism can also give rise to biases in value predictions. And so now we can define model 2, where the alpha plus and the alpha minus are proportional now to the slope of phasic dopamine responses, to positive and negative RPs. And so now what I want to do is just try to find evidence for either model giving rise to biases in value predictions in animals. And for this, we looked at a previous data set that was collected by our lab in mice. And basically, this study studied the avenula dopamine interactions. And the avenula, it's a structure that is upstream of dopamine neurons, and it's of interest to us because it is a major inhibitor in input-to-VT of dopamine neurons. And so basically, leashing it can modulate the level of inhibition that the avenula will have. And this modulation can be either on the scaling of dopamine responses or on the global activity level of dopamine neurons that could actually give rise to different changes in baseline dopamine. And briefly, in this study, mice were training a task in which different cues predict the rewards of different probabilities, and then underwent control or avenula lesions. And so if we now look at reward-seeking behavior and cue responses in dopamine neurons in the control animals, both of the readouts actually scale nicely and linearly with the probability of reward. So basically, scaling linearly with the expected value of the rewards that each cue is predicting for. And this is what we expect from an agent that is completely unbiased. But now actually we look at the lesion animals, the ones in red here, we actually see that there was an increase in the reward-seeking behavior and also an increase in the normalized responses of cue, in the normalized cue responses of dopamine neurons. And these features are actually something that we expect from a biased agent. So basically, an animal that is learning with asymmetric learning rates in favor of alpha plus, and it's kind of indicative of optimistic biases. We also observed this decrease in the magnitude of cue responses that I will kind of be explained by another intuition, but right now it's a bit counterintuitive. And so now, until now, this can be explained by either of the models I told you about. But now we'll try to look for basically signatures in the data and see what model is being supported by the data. And so first, we look at this model 2, where the asymmetric learning rates are a function of the slopes of the dopamine responses. And so for this, we estimated the alpha plus and the alpha minus from the slopes of dopamine responses, shown here in the population average for the control animals, and also at the level of each single cell. So basically, you can like derive this from each single cell and like end up with a distribution of asymmetric scaling factors. And an optimistic bias will be consistent with an increase in the slope for positive RPs and a decrease in the slope for negative RPs, as shown here. And in the distribution case, it will be consistent with a positive shift in the mean of the distribution. And so now what happens if we look at the lesion data that show optimistic biases? Basically, we didn't see any of the changes, like both at the level of population average and at the level of the single cell distribution, data was not consistent with the model 2. And so now let's look at model 1. Basically here, we're looking at baseline dopamine, so we look at baseline firing rates, because this could be an indirect readout of baseline dopamine concentrations. And so what we saw in the model in the data actually is that the lesion data presented an increase in the baseline firing rates. And in the model, this could lead to increases in baseline dopamine concentrations and lead to the regime where alpha plus is larger than alpha negative. And so now we can see what predictions this model makes and if we can actually find signatures of those model predictions in the data. And so the model predicts that with increase in baseline dopamine, there will be this increase in value predictors, both in the normalized case and in just the raw case for the model value predictors. And this is kind of like exactly what we saw in the reward-seeking behavior. That's kind of like a behavioral readout of the expected value. And also furthermore, in the Q responses, actually, the model predicted these two signatures that we saw in the data, basically, that there was this decrease in the absolute magnitude of Q responses and an increase in the normalized Q responses. And so taken together, model 1 was actually able to posthumously explain various aspects of the data that were observing lesion animals and these optimistic biases, whereas model 2 were not, like the signatures of model 2 were not found in the data, at least model 2 not being implicated in the emergence of these biases. Okay, so yes, to conclude, we proposed a model for biases in RL that highlights the importance of baseline dopamine as a candidate mechanism for generating biases in model predictions. And this model makes use of the opposite effects of baseline dopamine on the receptor sensitivities. And it could possibly explain various aspects of the data in albino lesion animals. And finally, this mechanism can actually be linked to findings of altered baseline dopamine levels in various psychiatric conditions and also some pharmacological effects. And with this, I conclude. Thank you for listening to me, and also thank you for all the members of the Shia Lab, especially. Yeah, that's all.
Any questions from the audience? There's one from the online session. Question from Kevin Lloyd. I was wondering how your model relates to the OPAL model of Van Collins and Michael Frank.
Yeah. Yeah, it's quite similar. I feel like in the OPA model, they're using this difference. It's also in the softmax function for eliciting for action choosing. So it's more like, I feel like that model was like, could be better translated to performance issues more than to learning aspects of it. But it's entirely compatible with it in the end. It's just like the OPM model is not really telling you how an asymmetry between these two populations could happen. And this model is kind of like trying to find out why in the brain this could be happening.
Okay. All right, I don't want to be too pessimistic about our timeline, but since we're running a little late, let's...
Yeah, sorry.
Thanks, Andrew, again. So we have 2 more contributed talks before 2.5 hours of lunch break. Our next speaker is Chengguan Li, if they're here. Yeah. Okay, she's coming. Her talk is on integrating deep reinforcement learning agents with the C. elegans nervous system.
Can everyone hear me? Okay. I'm Chen Guang. I'm here today to talk to you about integrating DeepRL with the C. elegans nervous system. So DeepRL has been extremely successful in a wide set of domains. And here in our project, we wanted to see what would happen if we integrated it with a living nervous system. And specifically, we wanted to see how an artificial neural network could be interfaced directly with a biological one to enhance the biological network for specific tasks. We chose the nematode C. elegans because of its simplicity and wide array of genetic tools, and we chose target finding as our goal. In this talk, I'll first go over how we built the system, from the animal engineering to the interface between AI and animal, and then describe some details in the AI itself. Then I'll show you how the system is performing in getting the animal to go to a target, and finish with some preliminary data on the ability of the artificial and biological neural networks to compute information in a cooperative way. All right, so throughout this talk, I'll have references on the side here if you're curious. We modified the animals with optogenetics. And here we first inserted channel rhodopsin in a specific sensory neuron called AWC-on. And that neuron activates when animals turn away from attractive odors. So in practice, when blue light flashes on the animal, it'll reverse or go backward. And this is a different sensory neuron called ASH, but it has similar behavior. So you can see generally what the animal's movement looks like. In this video, it's sped up a little bit. To connect the animal to the computer, we used a light that the computer controls and a camera that watches the animal and then sends images back to the computer. The images are processed in real time before being fed to the AI, 15 frames at once, and each frame is processed into the coordinates of the animal on the plate and the sine and cosines of the head and body angles relative to the positive x-axis. So for 15 frames and 6 variables each, that's 90 variables that comprise one input that the AI sees at each time step. For the AI itself, we chose DeepRL. And RL, like in the previous talk, it maximizes a reward signal over time. And for us, that reward signal was the proximity of the animal to a given target. The agent can see this reward signal and observations from the environment, which for us was the animal and its surroundings. And the agent takes actions by flashing the light on or off on the plate, forming this closed loop system, and then it learns from the environment throughout training. In engineering the AI, we had a few challenges to address. First, we had to make sure that the algorithm was flexible to different neural connections. We also had to make sure that it could learn a good temporal sequence to activate neurons in a pretty high-dimensional search space. And these first two issues could be solved by just choosing a good algorithm. We settled on soft actor critic because it's known to be fairly stable and also work in real-world applications like robotics. We also had to handle how data-greedy RL can be. So in the first DeepRL papers, it took millions of hours for agents to learn Atari, and that's not ideal for a grad student. So we had to do some offline training and first record a pool of 5 hours of data where the light was flashing randomly, and then train agents on that pool during development. We also augmented our data through random translations and rotations, and both of these things helped our efficiency quite a bit. The last thing I'll mention here is that RL can be quite unstable. So even after an agent learns a good solution, it can forget it after just a bit more training. And we tried a few different regularization techniques for this, but what ended up working best in the end was ensembles. 20 agents were trained on the same pool of data, and through randomness in initialization and training, they all learned slightly different solutions to the same target finding task. We put them together in a pool and had them vote on actions during test time. So in summary, the agent is an ensemble of soft actor critics trained offline on 5 hours of animal recordings with data augmentation. And we trained it to navigate animals to targets. And now I'll show you results. So this is the basic evaluation setup. We put the animal in the middle of the plate and randomly chose one of these 4 targets for the agent to send the animal to. When we tried this on the AWC on animal, it worked. With the agent, animals got significantly closer to the target than without. And below is a sample track. Animals start out at the black arrow, and the target is on the right. The dark green parts of the track are where the agent turned the light on, and the big circles I've highlighted are regions where the animal has just started to turn away from the target, making the agent turn on, and that's exactly the kind of behavior you would expect from a neuron that makes the animal reverse. To test robustness to neural variability, we tried 2 other genetic lines. Line B excites an interneuron known to be involved in chemotaxis called AIY, and we wanted our agent to not only be able to learn on single neuron connections, but also multiple neuron ones. So line C inhibits a kind of grab bag of sensory neurons, interneurons, and motor neurons using a particular promoter. Agents were able to learn target navigation on all of these lines, though the behavior for line C was slightly weaker than the other 2. Agents also learned different strategies, so they tuned themselves to the neurons that they were connected to. And we could see this by looking at the activation patterns on the tracks, or by studying the agents themselves and the weights that they learned during training. And finally, I'll show you some preliminary data we have on the ability of the animal and agent to compute information cooperatively. And we want to distinguish this between other more one-way interactions, like in brain-machine interfaces, where an animal is in complete control of a machine, or in this soft-living micro-robot paper published last year, where the authors turned a C. elegans into just a robot, controlled by a computer. To do this, we designed 2 experiments. And in the first one, we put the animal on a plate with paper obstacles and put the target in the center. We wanted to see if the agent could handle big picture tasks, like getting the animal to the target, while the animal handled the smaller things, like using its own sensory system to navigate around the obstacles. In the second experiment, we put food on one end of the plate and the animal on the other, and put the targets at increasing distances away from the food. In this case, we wanted to see if the animal's food search abilities could be enhanced, even if the targets were misplaced. And at some point, the animals have to decide whether to keep listening to the agent, which is telling it to stay at the target, or to listen to its own sensory system, which is telling it that food is somewhere else. And in both of these tasks, I want to stress that we didn't retrain agents on either of them. So agents have never seen data before with obstacles or food before test time. These are some sample tracks for the obstacle experiment. With the agent on the left, the animal started out in purple and then made its way over to the target in 20 minutes. And without the agent, it just kind of wandered around for a while. These are the statistics for the sensory neuron and interneuron lines, and both of them were significant, depending on your p-value cutoff. So the fractions are the fraction of animals that made it to the target over 20 minutes. And I think this shows that it does seem like agents can improve performance on these obstacle tasks without explicit training on them. Finally, in the food search experiments, again, these fractions up top are the number of animals that succeeded over 20 minutes. In this case, success means making it to the food. Without the agent, on the right here, no animals made it to the food. And with the agent, success scaled with the target's distance from the food, showing that the animal's food search abilities were enhanced. And it seems like in both the obstacle and the food search cases, the agent-animal system is computing information in a cooperative way. With that, I'd like to thank my advisors, Gabriel Kryman and Sharad Ramanathan, for just being really great throughout grad school so far, and also the people in both labs, Tim Hallisey, Abdullah Yonar, and Surya Bhupati Raju, for helping me get through the C. elegans experiments to start, and discussions about machine learning research along the way. Okay, thanks.
All right, go ahead.
Really fascinating talk. There's a concept in brain-computer interfacing that people talk about called adaptation, where the brain is adapting to the machine and the machine is also simultaneously adapting to the brain. So I'm curious what your thoughts are in your setup about that.
Yeah, so we train everything offline because of the just data efficiency issue. And I think a natural next step would be to train everything in the loop. I do think that the RL agents, it's very likely that they would be able to adapt with the animal, but there's also these issues of instability and forgetting that could happen too. So I think that will just take some more just studying of regularization in deep RL and figuring those tasks out along the way.
Fascinating talk. Hi here.
Oh.
I was wondering, have you also considered if the RL agent can help to train the animal such that later on, even without the agent, it can perform tasks better than before?
Yeah, so with the food search experiments, it is helping the animal find food. And I think that, again, so all of these episodes were quite short. They were 10 to 20 minutes long. But there would definitely be adaptation, especially with the sensory neurons, if we trained animals with this for longer periods of time. I think that's an interesting question that we would want to look at later, is adaptation and how the animal might adjust to the rewards given by the agent.
Thanks.
Mm-hmm.
Maybe I'll ask a quick question. Since you're controlling the animal's behavior, do you require the animal to have a sufficient amount of variability to be able to execute these policies? And if so, does they learn, the agent who's learning this, do they exhibit such features?
Yeah, so if you look at the video, the animals are pretty random. We never really can get them to do exactly what we want. So just inherently in that, because of the activity of the rest of the nervous system, it's a quite nice kind of inherent source of variability that the agents are learning from and exploring in.
All right, let's thank her again. Okay, the last talk of the session is by Adi Nair from Caltech. The title of his talk is Dynamical Systems Analysis Reveals a Novel Hypothalamic Encoding of State in Nodes Controlling Social Behavior.
All right, okay. So we're going to switch things up a little now because we're not going to talk about learning anymore, but rather innate behaviors performed by animals that are essential for their survival. Because at the Anderson Lab, we care a lot about how motivational states are represented in the brain. States that can cause behaviors like mating, fear, and aggression. And what we want to know is whether or not there's a common neural architecture that supports the encoding of a scalable and persistent motivational state. Scalable in the sense that these states can exist along a continuum. You can either be slightly angry or really angry, and persistent because once you become angry, you tend to stay that way for a little bit of time. And over a decade of research is focused on the hypothalamus as being a key mediator of these motivational states. The hypothalamus is a deep brain structure and consists of dozens of small little nuclei, and none of them receive a lot of love here at Cosyne, but it's got a very remarkable property. If you activate small groups of neurons, genetically defined, for example, estrogen 1 receptor expressing neurons in the portion of the ventromedial hypothalamus called the VMHVL, you can elicit really remarkable kinds of behavior. For example, if you activate this population, you can evoke aggressive behavior in male mice, where mice will relentlessly bite and chase their conspecifics. The VMHVL has a counterpart in a structure called the medial preoptic area, which is a structure that controls mating. And if you activate ESR neurons in this structure, you can elicit mating behavior in male mice, where mice will start mounting females and sing in the form of ultrasonic vocalizations. The fact that you can elicit these pretty phenomenal behaviors by activating small groups of neurons in the hypothalamus has reinforced the notion that the hypothalamus is perhaps just a labeled line. It contains these behaviorally tuned and transcriptionally unique cell types with very distinct downstream projections, and that perhaps there's not a lot of interesting recurrent computation going on. But a lot of this evidence has come from perturbation studies and not studies looking at representation at single neurons. And to do that, Tomomi Kurigo, a postdoc in our lab, obtained neural recordings from either the MPO or the VMHVL in freely moving animals using a head-mounted microscope. And so we recorded GCAM success signals from about 100 neurons or so. And if you look at cells in the MPO, you can find several neurons that are tuned to various aspects of mounting behavior and mating behavior. If you look at this a bit more broadly across all the neurons, you can construct a simple behavioral tuning raster. All the columns here are activities of individual neurons, and the rows are their activity during different behaviors that we've annotated for, organized as either female-directed or male-directed. And from the plot, what you see is that there are a lot of different clusters in the MPO. And if you focus on these two rows, you can see that neurons in the MPO are tuned towards mating behavior. This very much fits in line with what you'd expect from a label line view of the structure. But if you look at the ventromedial hypothalamus, the structure that controls aggression, you don't actually find neurons that are tuned towards aggressive behavior. Neurons are just active during the entire interaction bout. And if you do the same kind of analysis, you find that the majority of neurons are in fact tuned to the sex of the intruder. And in fact, if you look for neurons tuned towards aggression, like behaviors like attack, you really just don't find any. And this brings up a kind of paradox that you've seen in many neural systems, where perturbations reveal a representation of something like aggression. When you look at the imaging data, you don't really find neurons tuned to those specific behaviors, and suggesting that it may not be a simple label line as we thought. And so to resolve this paradox, we took a look at population dynamics. And our approach was this. We wanted to fit a dynamical system to our neural data. And the model that worked best for us were a class of models called recurrent switching linear dynamical systems, developed by Scott Linderman at Stanford. And the model works by, instead of fitting the activities of individual neurons, we work in the neural state space by looking at the latent factors that we first initialize using factor analysis. We split up this latent factor space into several different discrete states. And by fitting a dynamical system into each of these states, a linear dynamical system, you can effectively approximate a nonlinear system. The model switches between these different linear dynamical systems in a manner that recurrently depends on the population vector at any time point, hence called an RSLDS. There are two different things we need to optimize for here. One is the dimensionality of our latent space, and the other is the number of discrete states we need. And we optimize this individually in every animal by looking at the log likelihood of our fits. On average, we need about 7 to 8 dimensions in our latent space, and we need about 3 to 4 discrete states. So what can dynamical systems tell us about representation in the hypothalamus? So here's an example of a recording from the ventromedial hypothalamus. This is about 20 neurons recorded for more than 300 seconds. This is from a male mouse that's interacting with 2 different male intruders. And when we use RSLDS, we can identify several different discrete states, which are very long-lived. To figure out what these states mean, we can compare them to the different behaviors that we annotate for that the animal performs, behaviors like sniffing and attack. And we find that there are states that are pretty composite and consist of different phases of a motivational encounter. We have a state where mostly sniffing takes place. We have a state where we have this blue behavior called dominance mounting. That's a lower-level form of aggression. And we also have a predominantly aggression-enriched state, revealing a surprising representation of aggression in the population state space, even though single neurons themselves are not tuned towards attack behavior per se. But because we have an RSLDS, we can dissect this further and take a look at the different states and figure out how the model works. To do this, we took a look at the eigenvalues of the states in our model and converted them into a time constant to reflect the persistence or the stability of the different modes of our system. And when we did this, we found a single large time constant in all our states in every animal. And the dynamics of this time constant reflected that of an integrator. It had a low amount of activity at the beginning of the trial when the animal was showing mostly sniffing behavior. It ramped up over dozens of seconds when the animal displayed dominance mounting, this low-level form of aggression. And it remained persistently high when the animal displayed attack behavior. And we could find this in every animal. All the dots here are different animals. Surprisingly, if you look at the variation of the value of this time constant across animals, it also varied quite nicely for the fraction of times the animal spent attacking, which is a proxy for the animal's aggressiveness. And so what we've identified here is an integration mode in the VMHVL that seems to encode a kind of aggressive motivation drive. We can also ask how things look in a much more reduced dimensional state space. To do this, we use PCA to find the first 2 principal components of our high-dimensional latent space. And to visualize our dynamical system a bit more easily, we converted the flow fields of our system into a pseudo-energy landscape, where the length of a vector at any point reflects the height of the energy landscape. And what we found were these trough-shaped landscapes in every animal of the VMHVL. Different aggressive behaviors are found in the different stable points along the trough. And as you moved along the trough from one end to the other, you express different behaviors. Sniffing on one end, dominance mount on the middle, and attack on the other. The dots here are, again, from different animals. And thus, we find that the V-image field possesses these really slow dynamics and persistent activity. And in our model, this is implemented as an approximate line attractor. And so RSLDs helps us uncover a representation of an aggressive state that's implemented as an approximate line attractor. And this is pretty exciting because this can encode for both the scalability and the persistence of that state. But we also wondered whether or not this might be just an arbitrary fact of our fitting process, or maybe if you look hard enough, you can find an integrator in every hypothalamic structure. And to verify this, we did the same analysis in the MPO. The MPO is a structure that controls mating. And cells in the MPO have much more transient patterns of activity. You can see here, again, 20 neurons for more than 600 seconds. And we have this is a male mouse interacting with two different female intruders in this case. And RSLDs here finds very different states, much more short-lived, and states that transition very quickly between some cells. But if you compare the states with the actual behavior, you can find a really close correspondence. So state 2 is where you have mostly mounting taking place, and state 2 is where you have mostly intermission taking place. Hence, our model, again, discovers a representation without any reference to the actual behavior annotations that the animal performs. When we take a look at the actual states and the eigenvalues of our states, surprisingly, we don't really actually find an integrator mode in the MPO. And if you look at things in a reduced dimensional space, what we find are these rotational dynamics during different mating bouts. Each of these trials are when the animal displays a different mating episode, where the animal displays sniffing, followed by mounting, followed by intermission. And you can see that this is followed by a sequence of cells being active. And the rotation here simply corresponds to the activity of different neurons along different angles of that rotation. If you quantify the angle of the rotation, it actually varies quite easily between sniffing to mounting to intermission. And so what we found here, we think, is that our two distinct coding schemes for encoding very similar kinds of behavior, mating and aggression, in the V-image field versus the MPOA. In the MPO, we can find an encoding of mating actions implemented by sequential activity and rotational dynamics, versus in the V-image field, we can find an encoding of a motivational state, where the scalability and persistence of that state are encoded by an approximate line attractor. And this is quite exciting to us because these pairs of interacting GABAergic and glutamatergic nuclei are actually plenty in the hypothalamus. Another example of this is a structure called the AHN and the V-image DM. They both control fear behavior, and we hypothesize that the glutamatergic structure here may have a representation of a fear state implemented as a line attractor, whereas the AHN might have a representation of fear-related actions. And with that, I'd like to thank my mentors, David Anderson and Nan Kennedy. Tomomi Kurg is a postdoc who collected all the GCAM success data you've seen today. Bin Yang is a senior grad student who contributed some GCAM 7F data to the work. Scott Linderman helped us a lot with the modeling work, and Scott Ganguly and Mark Schnitzer were really helpful in conceptualizing this project at the very early stages. And with that, I'd like to take your questions.
We have time for some questions. Well, I'll start off with one. It was a fascinating talk. I actually have too many questions. Let's see. So the final model that you're proposing here, how does it resolve the paradox you presented earlier about the representation and perturbation differences?
Yeah, so it's interesting. So when you look at the single cells, you have an encoding what looks like sex. But what you actually have in the end is that you have these cells that are tuned towards either the males or females, but you have two different orthogonal line attractors. Towards males, you have a line attractor that encodes an aggressive state. The same structure also contains a line attractor that encodes a mating state. And so what this looks like sex encoding is actually much more complicated. They actually encode seemingly these motivational states, these orthogonal axes in different populations. And so that's what we think.
I see. Cool. Question over there.
Hi, thanks for the talk. It was really interesting. I'm just wondering if you have any ideas for mechanistically how the attractor landscape changes, maybe over the course of a session or in response to various stimuli.
Yeah, that's super interesting. So we've actually have a study in the lab that's shown that if you expose a male mouse repeatedly to a male mouse, they get more angry over time. And they tend to remain angry for even longer periods of time. And that's mediated by plasticity from a synapse in a structure called the AHIPM to the structure, the ventromedial hypothalamus. And we've actually looked at some energy landscapes in such animals, and it basically gets deeper and deeper, and it gets tilted in a way that you tend to stay in the deeper part of the attractor. So I think there is a really cool way in which you can link plasticity to different aspects of the energy landscape. So yeah, it's pretty interesting.
And is there an understanding of like the morphology of, say, the postsynaptic cell door, or like, you know, what exactly is going on at the synapse?
Yeah, so we don't really know, and the thing here is that the persistence we see is like really long, and we're talking about hundreds of seconds, not like the kind of persistence we often talk about in the cortex. So we've had a modeling study where we've shown that you actually need neuromodulatory input in addition to recurrent dynamics to support this. So we're currently now seeing if you can, let's say, knock down different neuromodulatory receptors, say like oxytocin or vasopressin, and if that suddenly, you know, takes away some degree of the persistence, and that would be really cool.
Great, thanks.
Thank you.
All right, let's thank again for the great talk. All right, now we have the lunch break until 3 o'clock. Our next session will be on spatial memory and beyond, so enjoy your lunch.